Thank you for those that have joined and those that are still joining. And welcome to our webinar that we are running by Central and South Genomic Medicine Service Alliance. If we can just go forward. So just to let you know that that the webinar is being recorded and it is being shared, but it is also going to be uploaded onto our website and onto our YouTube channel. And actually on the YouTube channel, you'll see this is the second one that we're running. Our first one that we ran in September is already on the YouTube channel. Can I just ask you all to ensure your camera is off unless you're speaking, and happy to show your face whilst you're talking. And can you also make sure that you are on mute? We have had a discussion amongst the team, and we thought it's probably best if we have questions. If anybody has questions, if you can add them in the chat, and then what we'll do is at the end, if the speakers are still available and happy to stay to the end of the meeting, if of the webinar, if we can go through the questions at that point, and that's just for time purposes. So if we can go to the next one, please. So I think there's much more people now. So thank you all for joining. I am Phil Costello. I'm one of the lead genomic nurses in the Central and South Genomics Medicine Service Alliance, and joining me today is our nursing and midwifery team. And we've got Charlotte Hitchcock, who is the Associate Director of Nursing and Midwifery. We've got Donna, who is the other lead nurse, and Donna is based up in Birmingham. I'm based down in Southampton. Donna Gillen is our other nurse, and Donna is also working on our IT and doing all the slides for us. So thank you, Donna. And then we've got Karen Creed, who is our lead midwife, and Karen is going to be doing a fantastic talk for you as well soon at some point during this webinar. So if we can go to the next slide. Okay, so our agenda today, we hope that as I said, this is the second one. So what we are hoping is that you get a bit more of a deeper view into genomics. Our first webinar really was a basic overview. So we've got Jess Williams, who is the Principal Genetic Counselor, who's going to give us some information on the basics of genomic biology. And then we are going to have Kate Glover from the Genomics Laboratory Hub, who's going to talk to us about some testing technologies. Karen, our midwife, is going to talk to you about cystic fibrosis, and then we'll have a break. Which for any refreshments. Sorry, can I ask that you can?
They've got funding, haven't they? So Alex has spoke to Tracy about giving me an.
Sorry. Then we'll have a talk from Felicity Blair, who is the Regional Lynch Nurse Syndrome CNS for Central and South Genomics Medicine Service, to talk to us about Lynch syndrome. We've got Michael Carter, who is the CNS leading the genomic practitioner team at Wessex Clinical Genetics Department, to talk to us about the nurse's perspective of whole genome sequencing. We also have Professor Sarah Ennis, the Professor of Genomics at University of Southampton, who is going to give us some information on genomics data, ethics, research. And then Hayley Wickens, who is our consultant pharmacist for the Central and South Genomics Medicine Service Alliance, talk to us about pharmacogenomics. And then hopefully we can do our questions. So that is our agenda today. If we can go to the next slide. What we're going to do, just start off, is just give you an overview of our first webinar that we held, and Donna is going to give us some information on the feedback that we received. So over to you, Donna.
Thanks, Phil. So as you all know, we keep sending these requests for you to fill in surveys. We've got a baseline survey for prior to the webinar, which is in the chat. Hopefully you can click on that and be able to fill that in. It's very short. It's only, I think, 5 questions. And then we have a feedback questionnaire at the end. And the purpose of that is really to make sure that we're aiming these correctly and that we're fulfilling the need out there around what you guys need to know about genomics. And obviously we're not going to hit everybody's level all at the same time, and we have advertised these as quite introductory. sessions. So I thought it'd be useful just to look back and see what we were told last time with the first webinar in September. And this slide just gives an idea for 2 questions. One, which was, will the information shared influence my clinical practice? So whether the people that came to the last webinar felt that what they'd learned would potentially have an impact on how they cared for their patients. And as you can see, we've mainly got blue, which is 56% said definitely, 11% said probably, and 33% said maybe. So some aren't quite converted yet, but are prepared to come and listen. I take it as a good message that we had nobody say no, so that was good. So I think that's we're on the right track, but maybe could do better, which is why these surveys are so important, so you can tell us exactly what you want to hear about. The second question up there is, have my educational needs been met? Again, the majority said yes, 44%, 34% said somewhat, and 22% said mostly. So again, good majority, but potentially we could do better. So keep feeding back, keep telling us what you want to hear, what you'd like to know. I'm just going to put this one up as well. We did ask specifically what people would like to learn about. And obviously we've got midwifery up there. So we have Karen speaking today. And we've got plans for other sessions that will be specifically for midwives. So keep watching our feeds. We've also got, there was a request for Lynch syndrome. So we've also got Flick, one of our specialist nurses, who will be talking about Lynch syndrome today. We'll have information on the test directory. We've got Kate Glover from the GLH who'll be talking about that. Hopefully demystifying the jargon of genetics will run right the way through everything we're doing. You'll have to get your own coffee and popcorn, I'm afraid. And then Jack, that little one up in the corner, that's the Genomics education programme. We're completely linked into that programme. There'll be various prompts through this webinar to point in that direction. And we'd encourage all of you to go online and have a look and see. And then just to mention, there's a little note there about health economics. The next webinar, which is going to be in February, I think the 28th off the top of my head, is really going to focus on the patient. 29th, was it? It's really, oh, that's right, leap year, caught me out. We're going to focus on the patient experience. So we'll have patients coming along to that one, speaking, and one of our professors in health economics is going to talk about insurance and genetics. So that will be coming. So I suppose this is a plea to say, keep telling us what you need. If there's anything specific locally you need as well, there's an option in the feedback form to send us your information, what you need to know, and we're happy to link in locally with you. So we are listening and we are trying to give you what you want. Okay, so I hope you have a great day today. Thanks, Phil.
Thank you, Donna. Yes, so really the feedback is very important for us and actually helps us tailor these webinars that we hope that we can get more genomics awareness and knowledge out to you all. But also through this webinar as well, Donna is kindly going to put together some questions. And so you'll have an opportunity to also test your knowledge as we go through. So I know we're slightly running ahead of time, which is good. So I'll probably, if we go on to the next one, if Jess is ready, what we'll do is just dive in now and go on to Jessica Williams, our principal genetic counselor, who's also one of our education and training leads within the Genomic Medicine Service Alliance, who is going to do a great talk now on the basics of genomic biology, just to get you all into the mood of what to follow. So over to you, Jess. And are you going to share your slides? Are you happy to share your slides, or do you want us to move them for you?
Would you mind moving them for me, please, Phil? Just because you've got all the doozy double helix down the side and I always put nothing on. I'm rubbish at that.
Well,
my skill set.
Thank you. I'm going to pass that on to Donna. So Donna's doing all the technology.
That's fine. You just prompt.
Is that okay? Thank you so much, yeah. Donna. I'm just going to talk a little bit about the basics of genomic biology, and I understand that kind of your groups of midwives and nurses, and I try to do this from the perspective of what a patient might ask you and how you might explain it to patients, which is essentially what I do pretty much all day, every day. So, Donna, next slide, please. So I'm going to go through kind of what is genomics, what is genetics, what's the difference? Are they important for all healthcare professionals to know about? And I mean, you voted with your feet in the fact that you're all here. So I assume that you think that genomics and genetics is important, but I wonder whether many of your colleagues do. And from the feedback that Donna gave from the previous webinar, it does appear that there's a little bit of a mountain to climb for kind of bringing on board other members of the team, perhaps. So I think that the thing that has always kind of made me realise that genomics is very important and it's important to patients is that has anybody ever asked you, does this run in my family? Will this run in my family? Could this affect other people? And why has this happened to me? And sometimes we can answer those questions for people, but I think that as general public awareness is getting greater of genomics and genetics, I think that this question will be asked more and more. And I know that you mentioned that there's going to be talks about research, but just this week, the UK Biobank released the information that they were sharing everybody's genome, so that researchers can go into the UK Biobank and maybe they want to study, I don't know, high blood pressure or why some people are short-sighted, all sorts of things, and they will be able to access their genome for research purposes. So research is going exponentially high. So it's very current in the general kind of media forums, I suppose. Next slide, please. So this is going right back to basics, and this is how I would explain genetics and why it's important to my patients. So the thing that I would say is that all living things have a genome, and within our genome, that's our DNA, and those are the instructions that tell our bodies how to grow, how to develop, and how to function. And the way that I describe it to patients is I would say, we all start out as a single fertilized egg. And how is it that that egg, that one cell, knows how to divide and then to develop into an embryo that develops into a baby that's born, that grows into a child and then into an adult? How does a bone know to grow to be a bone, which is very different to a heart growing into a heart? And it's our genes that do that. They give it the instructions that tell our bodies how to do all of these things. Next slide, please. So we can't see our DNA, we can't see our genes, but what we can see are our chromosomes. So I don't know if this is something that you're all familiar with, but we all have 46 pairs of chromosomes, and males have an X and a Y and females have two X's. And again, how I would describe this to a patient is when you see on the science programs, the double helix that's always at the start of a science program, if you squashed that right down, that's what your chromosome is. So your chromosome is essentially just squished down DNA. Next slide. So this is the thing that is starting to get very interesting for people, in the sense of, for most people, so for about 99% of our genome, it's the same, or 99.9% of our genome is the same as everyone else's. But it's that 0.1% that equates to about 3 million differences That's what makes us different. That's what makes us individuals. However, what we don't are trying to understand is whether those differences actually cause any health issues. Next slide, please. So again, this is something that I think is always quite interesting for patients to know and understand, and it also helps us understand when we are looking at trying to understand whether genes have stopped functioning or not, whether part of our DNA is giving a really important instruction via a gene, is that we share, you know, a significant amount of our genetic information with different species. So we share kind of 96% with chimpanzees, about 90% with mice, and about 60% with bananas. Now, the reason that that is quite interesting for us is because if we see a gene, if we find a gene or a section of DNA that is what we call highly conserved across species, so we know that that gene hasn't changed, whether it's found in a banana, a fruit fly, a chimpanzee, or a human, we know that that must be quite fundamental for life and for functioning. So that's why, although it's a fun fact, it's also quite an important fact as well. Next slide, please. So again, as I was explaining, there are some differences in our DNA that determine physical characteristics such as our eye colour, and others will influence our susceptibility to develop different types of diseases. So the things that we always talk about and we were taught at school, and I bet loads of you, because you've all got the cameras off, are seeing whether you can roll your tongue or not, which was always said to be genetic. And it was one of the first things that when you were in your science class at school, when they were talking about genetics, the teachers would have asked you to do this about whether you can roll your tongue or not. Really, obviously, eye colour, hair colour, whether you've got curly or straight hair, those sorts of things are determined by look, Donna can roll hers. I bet you did it as well, didn't you, Donna? So yeah, these sorts of things are determined by our genes, and some of those, but I suppose the important thing to be aware of is that there are other things that our genes determine, not just our physical appearance. And that's something that when I speak to patients and they'll say, oh, I know that I'm going to get this illness in my family because I look just like my mum. And it's about kind of resetting that perception that you really do get half of your genetic information from your mum and half from your dad. And just because you look just like your mum doesn't mean to say that you inherited all of her genes. And those are the kind of some of the common beliefs that families have, and that can be quite interesting to kind of help people adjust to that knowledge that chance is different to what they may think it might be. Okay, next slide, please. So I kind of feel that I've explained this, hopefully okay to you guys. So right in the center of our cells is the nucleus, and within that nucleus are our chromosomes. If you stretch that out, you get our double helix. And what we are looking for, particularly right now, is at our nucleotides. So the proteins, that code that gives those instructions that tell people how to grow, develop, and function. Next slide, please. So when we're looking at genetic variation within the genome, not all variants are pathogenic. And so we have to be very careful how we interpret that data and how we give those results. And the way that I kind of describe it is that for example, if you live in America, you spell words slightly differently. but it doesn't change the meaning. So I could go to America and I could read a book that has been published in America, and I would still be able to read it and understand it. Because although there are slight changes, it hasn't kind of changed the sense of that sentence or that word. So next slide, please. So as I said, that not what effect do variants have, and so sometimes they have no effect at all, and we would call those benign. They might have an effect on appearance, again benign. They might have a slight effect on health. So as we talk about a slight increased risk of glaucoma, and I'm pretty sure that most people, when they look at their family history, will see that there are some common conditions, so not necessarily genetic conditions, but there are some common conditions that affect people in their family. So they see it running in their family, and it may not be caused by one single genetic variant that we know can cause, for example, cystic fibrosis, but it may be a combination of environmental and other genetic factors. And then there can be a severe effect upon health, so a single gene disorder, which is what we class as pathogenic or disease causing. Next slide, please. So when I first started working in clinical genetics, which was a long time ago, what happened is that we would be referred somebody in, for example, somebody, maybe an individual who was having health issues such as learning difficulties, maybe some health problems, for example, seizures, and we would be asked to see those individuals to see if we could make a genetic diagnosis. And what used to happen is that we would do what we call a phenotype. So that's kind of how that patient presents. So it might be brain MRIs, it might be cardiac MRIs, it might be just how somebody physically looks. So we would get that kind of idea of how somebody appeared as far as regarding their health. And then the geneticists, the genetics team would think about what this might be. And so we would order that test. And then what would happen is we might get a result on the first test. If not, we might think, oh, well, we'll try this test, or we'll try that test, or we'll try and it might go sequentially on. That doesn't happen so much now because technology has changed, and because genome sequencing is becoming quicker and more cost effective, we are able to actually sequence people's entire genome in a relatively short space of time for a relatively little amount of money. The difficulty is interpreting that data. So basically we get the entire genome, and then the scientists, the bioinformaticians, the genetics team, their job is to try and interpret whether those variants actually have caused any health issues. Next slide, please. So I think I can kind of, the only thing that's important to know about this slide is that we have a reference genome, and it's not just one person's genome. So at the moment we're on build 38, but that reference genome will change over time. And one of the things that one of the aims of changing that reference genome, so what we compare it against, you know, an individual's, a patient's genome, it has to be compared against something to know whether there's any differentiation. And what we hope is that well, the aim is for that reference genome to become far more representative of a diverse population. So at the moment, it's completely overrepresented with the Northern European population, and that needs to change because it needs to reflect a global population. Next slide. So I'm going to, again, this is just how I would describe this to patients. I'm just going to talk to you a little bit about how I talk about how different variations have an impact upon the functioning of that gene. So I would describe it as what happens is you have a sentence that gives an instruction, and there's a change within that sentence that alters the function of that gene. Now we start with a missense, so you can see that the instruction is, or should be, the man saw his dog eat his hat. Now, sometimes there can be a single change within one of the code, and so you can see it goes from the man saw his dog eat his bat, or the man saw his dog eat his cat, neither of which a man wants to see his dog do. Next slide, please. So a premature stop, so all of our genes, they have to have a little instruction to start at the beginning and a little instruction at the end to finish. And sometimes we can get what we call a premature stop, which is a nonsense mutation. And so it stops too soon, and it's the man saw his, and you can see already these changes, they are stopping the gene from functioning as they should. Next slide, please. So a frame shift is when you get a small section of the code that is in the wrong place. So you can see from here, so where there has been a little extra bit added, I'm pointing at my screen, I don't know why, because you can't see me pointing at my screen, but you can see where this has been added on, and it's shifted everything along, and so it's just completely lost all sense. And that's just because a little bit has been snipped out, or it could be added in, and it just shifts everything along. Next slide, please. So when we look, and I spoke earlier about the variants that we can that we all have within our genome, we know that the vast majority are benign or lightly benign, so they don't really have any effect upon how we function, but just might have effects upon how we look, etc. Then there's another kind of section that what we call variants of uncertain significance or unknown significance, and that's when we can't make, or the bioinformaticians can't make that call of, has that gene change, has it stopped that gene from functioning as it should, or can it still function? And we can't make that call sometimes. And then you can see right at the bottom of this slide, there's a very small proportion of variants that are what we call pathogenic or lightly pathogenic. And that's essentially how variants are classified. They go from benign to lightly benign, unknown significance, lightly pathogenic or pathogenic. Next slide. Oh, I'm going over. Hold on, I need to speed up. Okay, next slide. Oh, perhaps I've finished. Next slide, please. So, oh. Okay, so thank you. So when we talk about single gene disorders, what we're talking about are the typical, when you have almost what we would call the bread and butter of clinical genetics. So that's when we're talking about when people have one single genetic alteration within a gene that stops it from functioning. And depending on whether whether things are passed on, whether you need two working copies or one working copy, can make an impact on how things are passed on. So I don't know whether people are aware of dominant, recessive and X-linked inheritance. So this is when dominant is when you have one working copy and one altered copy of the genes. So you have one normal copy and you have one pathogenic variant. And if you have that, you are expected to develop that genetic condition. Recessive inheritance is when you have one normal working copy and you have one altered copy or one pathogenic variant, and you are known as a carrier of that condition. And the time that it usually is significant is when you have children, because if two individuals who are carriers of a recessive condition, they have a 1 in 4 chance of passing that on to their children. And then there's X-linked inheritance, which is when women have so when the gene is on the X chromosome, so because women have 2 copies of an X chromosome, they tend not to be affected, but they have a chance of having a boy that is affected with the condition. And then mitochondrial inheritance. So the mitochondria are the powerhouse of your cell, and they have their own little bit of DNA going on. And so sometimes there can be changes within your mitochondrial DNA that can cause genetic conditions. Next slide, please. Right, I'm going over, so I need to. So multifactorial inheritance is when you have environmental factors and genetic factors. So you're born with your genetic factors that may make you more at risk of developing a genetic condition. And then environmental factors over time may mean that you develop conditions in your lifetime. That may be things like glaucoma, it may be types of cancer, it may be heart disease, all sorts of things. Okay, next slide, please. Sorry, oh, yeah, I kind of feel that I've explained that without showing the slide. So next slide, please. So the other thing that I think is going to be really significant for all healthcare professionals is the number of people who are having, particularly timely because we're coming up to Christmas, of people opting for 23andMe, so direct consumer genetic testing, and people will ask you about their results that have come through. And I think that it's very important to say that that what 23andMe or any of the commercial direct consumer testing companies, what they offer and how those reports are written and they're then interpreted by the people who take them on, who have ordered them, can be quite varied. So I think that it's open to interpretation, and it just shows that you need to have that understanding of this may or may not be significant. Okay, next slide. Really sorry about this, I'm totally overrun. So, the last one. So lots of tests can now be offered. It's about which is the best for the patient. So for example, there'll be some families where we can just test for that single gene, and there will be other people where whole genome sequencing is far better. So genetic testing is changing fast and it cannot be totally predicted. So always worth checking about possible testing for a patient, how genetic testing has changed over my time working in clinical genetics is phenomenal. And the importance of pre-test counselling. So people need to know that they're having a genetic test because this can potentially have impact on not just themselves, but other family members. So it's important that they know about that. And I think that's me. Sorry, I overrun.
Thank you so much, Jess. Don't worry. That was a great basics of genetics and genomics. I have put a question there, and hopefully, if you are able to stay with us, we'll come back to that question at the end. But thank you very much for that talk. Our next speaker is going to be Kate Glover, who's a scientist at the Genomic Laboratory Hub, who's actually going to give us more information, as Jess has already kind of segued into that about the testing technologies. So thank you very much, Jess, and welcome, Kate, and thank you so much if you are happy to start.
Yep, okay. So thank you for inviting me this morning. Hopefully I'm going to give you a fairly brief overview, I'd say, of whole genome sequencing, which has already been alluded to, so I'm not going to go back over that. But I think if I could have my next slide, it would be quite useful to just set out what I'm going to do for you. So very briefly, in hopefully 12 or 13 slides, depending on how we get on, I'm going to give you an overview of what a genomic laboratory hub is, or a GLH. There's lots of acronyms around genetic testing, so hopefully we'll be able to give you some background on that. Then I'm going to touch on the genomic test directory and specifically the eligibility criteria. So that is who within our medical workforce can actually request these tests on behalf of patients. Then I'm going to talk more specifically about the requirements for whole genome sequencing testing, or WGS. And that is because there is a whole separate set of requirements there. So we'll go over that. And then as a result of that, we'll touch on how we process those samples. And then my last slide contains a list of key contacts and website links. So there's lots of information in these slides, hopefully that you will be able to go back to later. And be able to look at your leisure, because I'm going to give you an awful lot of information that I'm not expecting you to retain. So if I could have the next slide, please. So the Genomic Medicine Service in England is divided up into 7 genomic laboratory hubs, GLHs, and they are responsible for all of the genomic testing across England. This is a restructuring of what was previously happening. I was trying to work it out. I reckon it's probably 5 or 6 years ago since this was kind of born out into reality. And the aim behind this was to provide standardisation for patients for all genomic testing across England. When we talk about something being a core test, that means that it's provided in all 7 of those GLHs and will be geography specific in most cases. And then other more rare conditions, or conditions where there's a degree of additional specialist knowledge required, those are divided up into 1 or possibly 2 areas across England who provide that specialist testing, and that's provided by certain GLHs. But from the perspective of ordering a genomics test, in most cases, your local genomics lab, so the one that you've always used, will be responsible for collecting the samples from the patients, and then they will send them on to the appropriate place for you to access testing for your patient. So if I could have my next slide, please. So this is an overview of what you will get if you click on that top blue link. This is the National Genomic Test Directory. And when you open this up, oh, if we could go back 1, that would be good. Thank you. You'll get 3 different boxes. The top one is the Excel, which we'll come on to in a minute. And that is literally a list of your condition and how we test for that, so to sort of direct how and which forms we need you to access for your patients. The middle box, the bit where it says rare and inherited disease eligibility criteria, is, I think, a more friendly document. It's a PDF, which gives you a bit more information. Again, I'll give you a snapshot of that in a moment. And just so we don't forget, I will be mainly focusing, I guess, on examples of rare disease genomic testing, but we do have a National Genomic Test Directory for cancer testing, and that has its own set of information, so that link is there if you're interested in that. So the next slide gives you a kind of snapshot of what's in that top box. So I'm not expecting you to read the detail here, but if you click the next animation, hopefully it will zoom in on one. So there we go, that's an example. You may hear things referred to as R codes, and that's R for rare disease, and each different condition or clinical condition or group of phenotypes is given a specific R number, and you will hear that talked about a lot and see that on different forms. In the cancer world, just so you know, those are M codes, M for malignancy. So this is an example literally just of a single R code there. So this is hereditary ataxia with onset in childhood, and you can see that it's broken down into different subsets, 0.1, 0.2, 0.3 and 0.4. you can also see, if you look across there, that the methodology we use to test for that, so that's WES, or large panel, which is whole exome sequencing, microarrays, STR testing, and then at the bottom WGS. So that's how you would at a glance see how we might in the laboratory direct the testing. But in most scenarios, if you collect the sample and send it on, the Genomic Laboratory Hub will sort out what needs to happen with that patient. So if I could have my next slide, please. The next one, which I think is more user-friendly, is the eligibility criteria. So this sets out who and which specialisms and which medics can actually order these tests. And the National Test Directory is continually reviewed. We're due another version, which will be coming out very soon. And if I can draw your attention just to the very bottom of this slide where it says requesting specialities, you can see there that those are the primary groups, I guess, of people who would request for this. And this is another example, SMA, something that you may all be familiar with. The R code for this is R70. And those requesting specialities at the bottom would be the groups of clinicians who would be most likely to request SMA. But it's really important to note that this is not prescriptive, and in most scenarios there is a degree of flexibility. We're starting to see, for example, with something like cystic fibrosis, that GPs can now access testing for some of these things. So it's always worth, I think, having a look at this document. Like I say, I think it's more user-friendly, and it's the middle link of the previous slide that I showed you. So I think the next thing on my next slide, I'm going to move on to whole genome sequencing. Oh, okay, I've got one in between. So just to finish off, I think I've said most of this already. How to request testing, you will have links to your local area and their local websites mainly for the request forms that are needed to access genomic testing. So that's for everything, every type of genomic test that you might consider. This is a bit of a plea. As a clinical scientist who receives in referrals, we get referrals on all sorts of different bits of paper, historical samples. Certainly in Birmingham, we always used to have blue and green forms, and those, I don't know where they're coming from, but they're still coming out. We haven't produced those for years. So I would just make a bit of a plea, I guess, on behalf of the clinical scientist team to use the most up-to-date genomic test request form. They've been designed for a reason, just to help everybody in the process. And most of those are available online, particular different websites. But for whole genome sequencing, there's an additional layer, and I'm going to explain why that exists. So there are additional forms, and potentially, depending on who and how you're referring your case in, that there are different requirements. And just also to say that hopefully on all of those forms, the requirement for those samples is listed. So if I can have my next slide, please. So in a nutshell, WGS analysis, certainly for most children, is in the form of a trio. And by trio, we mean mum, dad, and proband. And as a result of that, we need a certain set of additional forms to be filled in. There's a test order form, or a TOF, one of those per referral, and they must be now completed electronically. That's been mandated by NHSE, and there's multiple reasons for that, mostly because we want to make sure that we can read everything that's on that form. And then for each family member being consented, they need to have their own record of discussion. Something that gets lost, particularly with paediatric referrals, is that once a young person reaches the age of 16, they are responsible for their own record of discussion. Even if they're seen in a paediatric clinic, they still need to do their own form. And if an individual lacks consent, because they lack capacity to do that, then there's an additional need for a consultee form. All the forms, all the links are in this presentation, and they are all available online. So if we, in the interest of time, if we skip on to the next one, not going to go over the detail. This is what a whole genome sequencing test order form looks like. Again, a plea, all the boxes need to be completed. One of the major pitfalls we have is with the family members that come in and having 3 correct identifiers. So if you aren't able to supply that because you don't know or you're not sure, then it'd be really helpful in the relevant clinical information box to tell us that, so that we know how best to progress with this referral. All of this information gets uploaded onto a national genomics information system, or NGIS, you might have heard that word. And again, this is a nationally set out process and not one that we can influence. So that's the test order form. The next slide is just, I think, a high level view of the record of discussion, or the consent documentation. It's a 3-page document, and it's important that whoever is doing the consenting understands what they're consenting their patient for. So this is the first 2 pages, and then if you go to the next slide, the third one is the bit that we really need you to fill in. And if I can have the animation on the slide as well, thank you. So just some key points here. We need to make sure that on all 3 pages of the record of discussion, this information is transferred across so that we know that it all links together. The next animation, please. There's a piece there about confirmation of your genomic test and research choices. I will touch on that in a little bit more. I promise I've only got 2 more slides after this one. And the next animation, please. And this is the bit that quite often gets missed. So this is to be completed either by the patient or the family member. And then the bit at the bottom, the final animation, is completed by whoever it is that's having that conversation. So it can be the clinician, but in most cases, certainly within paediatrics and the clinical genetics world, that's done increasingly by genomic practitioners and specialist nurses who have been trained up to do that. So any questions, please ask on any of that. So next slide, please. I know there's a whole talk on the National Genomics Research Library, and I just want to draw out a couple of points here. Final point on this slide really is the important one, is that the patients, the families, do not have to consent to use of their data in this National Genomics Research Library in order to access diagnostic testing. Very often we find that it's not appropriate for that conversation to have happened, and people think that that may prevent their family from accessing genetic testing by whole genome sequencing, and it's not. So I know there's a talk coming later, so I won't say any more on this slide. And then the final slide, I think. Yeah, so this is just, I think I might have 2 more. Key point on this slide is that we have a dedicated GLH email account, and the email address is there. That is now monitored by an absolute army of fantastic administrators who are very, very skilled now with how we process these samples to get them submitted on behalf of the whole GLH. The standard route for those to come in is that forms are sent by email as PDFs. Bit of a plea that all paperwork should come individually rather than all scanned as one big document, because we have to separate it out if not. The TOFs and the RODs are all uploaded onto NGIS when we request that test, so it helps us if they can be sent across. As I've already said, we need fully completed forms, please. And I know that sounds simplistic, but if I told you that well over 70% of the referrals that we get in are still incomplete because we don't have the appropriate information we need. So I think my final slide, and this is it. This hopefully will give you some useful links, things that you can go back to. We produced right at the beginning of whole genome sequencing an FAQ guide, which I have not looked at for a little while, but most of the basics are there. So I think if you're interested and you want some more information on how to request whole genome sequencing, that's a good place to start, and the link is there. Thank you.
Thank you very much, Kate, for that. All great information. Just also to add that you can access those forms on the test order form, and the consent form is on the NHS England website as well. But thank you very much. The really clear explanation. I haven't seen any questions come in whilst you were talking, but hopefully we'll get some questions, and if you are available at the end, we'll go through them. Otherwise, I may send them on to you if there's anything that we need your expert advice on.
No problem. Thank you.
Thank you. So going on now, we've now got Karen, who is our lead midwife, who's going to give us a fascinating talk on cystic fibrosis. So Karen, on to you.
Hello, everybody. I'm going to take control of the presentation, Phil, if that's okay, or Donna, just so I can go through the slides myself. And just while I'm speaking, and I'll go off camera because I've got a bit of a glitchy thing going on with my IT and I don't want to cut out altogether. So Okay, let's just see. I've got control. Anyway, my name is Karen Creed. I'm lead midwife at CAS GMSA, and my background is largely in screening, fetal medicine, and prenatal diagnosis. So when I was asked today to come and talk about some aspect of genetic testing in relation to maternity care, I started thinking about what I was going to talk about and started reflecting on my career from back in the mid-80s as a student nurse, right through to being a student midwife in the early 90s, right through to current day. And throughout that time, I've witnessed how we've harnessed the power of genomics to improve our understanding of conditions, to develop new treatments, new screening programs. And in relation to prenatal diagnosis, I've seen that over time, how we've increased the number of options available to parents to support them in their reproductive decision making. So just move my slide on. So what I thought I'd do was talk you through a timeline of prenatal diagnosis or developments in prenatal diagnosis in relation to cystic fibrosis. And I've chosen cystic fibrosis, or CF, because it's a condition that probably lots of people have heard about. And I know that we've got a lot of non-midwives on the call. I'm also just going to mention newborn screening and a little bit on management of the condition, just to give you sort of other areas where our knowledge of genomics is influencing sort of management and care and so forth. And I'll have a look at some challenges for the future and signpost you as to where to find out more going forward. So Jess has already given us an overview of variants and how pathogenic variants cause disease, and she also mentioned the autosomal recessive inheritance pattern, which is relevant for cystic fibrosis. So it's a recessive condition caused by variants in the CFTR gene, and the CFTR gene provides instructions for making the CFTR protein, which in turn facilitates movement of molecules in and out of the cells. In people with cystic fibrosis, this process is impaired, and that results in the production of sticky, thick mucus that causes the clinical symptoms that we associate with cystic fibrosis. And of course, this affects the lungs in terms of persistent acute respiratory infections. It affects the GI tract in terms of abnormal stools, malabsorption, and malnutrition, and can impact fertility as well. About 1 in 25 of the Northern European population are carriers for the gene that causes CF. But if you're a carrier, it means you've got one CF gene variant, but the other CF gene doesn't have any variants that cause CF. So therefore, you wouldn't know you were a carrier unless you were tested. We don't have a national screening program in this country for CF screening, so most people are unaware, and usually the first time that they know they're a carrier is after they've had a baby. That can be quite a shock, and the only time that we generally test people is if they've had a baby or with CF, or if there's another family member with CF and they're concerned about whether they could carry the gene variant as well. So in an autosomal recessive pattern, as you can see here, we've got two parents, both carry one copy of a variant in the CFTR gene. And in any pregnancy, they can either pass on their healthy CFTR gene, or they can pass on the variant. And there's a 1 in 4 chance of having a baby with cystic fibrosis if they both pass on that pathogenic variant. On my next slide. So I just wanted to talk through some of the tests that we've developed over time. And I'm going to go back to the start of my career when I was a student nurse in the mid-1980s, so a very long time ago. And I was working in Birmingham, and we looked after children with cystic fibrosis. And at that time, it was largely seen as a disease of death in childhood. We hadn't identified any carriers at that point, so it was not possible to test for carriers, and therefore there was no prenatal diagnosis available to parents. So for parents, after they'd had their first baby with cystic fibrosis, all we could do was advise on the chance of recurrence in a future pregnancy. And parents could then take their chances and have another baby and hope that this time they wouldn't have a child with CF. Or in some cases, they might decide not to have any more babies. They could use donor sperm or adopt, but that was all we could really offer. Then in 1989, we identified the first variant within the CFTR gene that caused cystic fibrosis. And of course, once we start identifying variants, and that opens the door to prenatal diagnosis, or the potential for prenatal diagnosis. Initially, we found just one variant, but today we know that there's several hundred variants that cause this condition. We still don't know all the variants though, so we can never fully say that somebody conclusively will not have a child with CF. We can only reduce the likelihood because we can't test, we still don't know all the variants that are out there. And we have a way of collecting a sample from an unborn baby, and we can then analyse that sample and test it for the CF variants that couple might carry. And the way I would explain these tests to parents are that we use ultrasound scan to guide a needle through the lower abdomen into the womb, and with a CVS test would take a small piece of the placenta. With amniotic fluid we would take some, with an amniocentesis we take some of the amniotic fluid, and that will provide us with cells that we can analyse for CF gene variants. Not without risk though, so putting a needle in the womb of course can cause problems, and the risk of miscarriage from these tests is anything between 0.5 up to 1%. And then, of course, parents also have to think about what they would do with the information they would get from these tests. So it may be that they just want to know whether their baby has CF or not, so they can plan for having a baby with the condition, or maybe rule it out so they can enjoy the rest of the pregnancy and the knowledge that the baby hasn't got it. In some cases, some parents may choose not to continue the pregnancy, but clearly it shows that there's a lot of difficult decisions to make, and it requires highly specialised genetic counselling to talk through those options before you get to the point of having a test in the first place. It's worth mentioning, of course, that we use these same tests today for a whole variety of different genetic conditions, not just CF, for chromosome conditions as well, and we can even do whole exome and whole genome sequencing. And we are looking to put on a webinar about that next year, so if you're a midwife or working in fetal medicine, do have a look out for that next year. Next thing I just wanted to mention was pre-implantation genetic testing. So in the States in 1992, the first child was born free of cystic fibrosis following pre-implantation genetic testing. And in this process, and what happens is the parents go through an IVF procedure, and at about day 5 after fertilization, they remove some cells from the developing embryo and test them for the variants that we're concerned with, and only implant healthy embryos back into the womb. So this technology is available on the NHS, but it took a good 10 years. It was the early 2000s when it first became available, and it's available now, but there is strict eligibility criteria for who is eligible for these tests. Not only that, parents have to go through an IVF process, which of course takes time, and it's not always successful. But for some parents, what it provides is an option for avoiding having a baby with cystic fibrosis, especially if they wouldn't consider prenatal diagnosis from CVS and amnio and wouldn't consider termination of pregnancy. So moving on to current day, then we have a new testing process available called non-invasive prenatal diagnosis, and what this offers is a test that has no risk of miscarriage, and all we do is test some of the maternal blood, and in that bloodstream is some of the DNA that's come from the placenta. It's called cell-free fetal DNA, and we can analyse that for CF variants. Benefit, of course, is no risk of miscarriage, and it is available now in the National Genomic Test Directory. However, it does take, in some cases, we have to do preparatory tests, and these can take up to 8 weeks. So in an ideal world, we would counsel parents before conception so that we could do any preparatory tests before they conceive, and that can speed up the whole process. It's also early, we can do it earlier in the pregnancy, we can do it from 9 weeks. So there are challenges for getting women into care early enough in pregnancy, because of course some of these tests are time critical. We can now do NIPD for a whole variety of conditions, and if you want to know more, go and have a look in the National Genomic Test Directory. It lists all the conditions we can offer this technology for, and also again we're going to put on a 2-hour webinar early next year just to talk about NIPD in more detail. So again, if you're working in fetal medicine, do have a look out for that. We will advertise it next year. A couple of other things I just wanted to mention.
First of all, newborn screening. So we currently offer screening for all newborns on day 5 of life. And since 2007, we have added cystic fibrosis. So we screen for about 9 or 10 different conditions now, depending on where you are in the country. And CF has been there. The idea being that if we can diagnose babies before they become symptomatic, then we can improve the outcome for those children. With the blood spot program, what we're doing is looking for the downstream effect, if you like, of having inherited 2 CF variants. And we're looking for a raised level of IRT in the blood, which is raised for about the first 8 weeks of life in a child with CF. Coming next year is a new research program, and this is called the Generation Study. This is research, so it's not a national screening program, but the aim is to screen 100,000 newborns to look for over 200 medically actionable conditions, and this includes cystic fibrosis. But of course, this is screening in a different way. We are looking for cystic fibrosis variants. So whilst newborn blood spot screening will detect babies, or has the potential to detect babies who've got cystic fibrosis caused by a variant that we don't yet know about, with the Generation Study, we're only going to look for known variants. So for any midwives on the call, what's really important to be aware of is that the newborn blood spot screening program is more important than the Generation Study in hospitals where we're offering the Generation Study. So again, I've just put the link there to the Genomics England website, and they've got more information about the Generation Study. Genomics England are leading that program. So just a couple of things I wanted to mention about management and treatment of cystic fibrosis. So over the years, certainly from the start of my career, a lot of the treatment really has focused on that downstream effect of inheriting 2 CF variants, so managing sort of respiratory support and nutritional support. But there's been a lot of research taking place into new treatments, and the one I just wanted to mention was the group of drugs called CFTR modulators. So this is interesting because what it does is, rather than focusing on the effects of that sticky mucus, it's going in higher up the pathway, if you like, and it's targeting the defect in the CFTR protein to enable it to work properly. Many people receiving these drugs are reporting life-changing effects. It's not a cure, it's lifelong therapy, and there's still many challenges with the CF diagnosis. But what they're predicting now is that for some children starting this therapy today, then they may have near normal life expectancy going forward. However, there's a couple of caveats to this. It's not available to all patients with CF. So there's about 10% of patients who have variants, CF caused by variants, for whom this is not effective. And so when we're thinking about prenatal diagnosis, for example, it's important to know what gene variants parents have, so that we can accurately give them information about what treatment might be available or not available for their child. Secondly, CFTR modulator drugs have been in the news recently, and you may have seen, it's been in the general media, that there is some concern about the cost of these medications for the NHS, and that it's currently going through a consultation process with NICE. So more information is expected on that later this month. So it's a little bit of an uncertain time at the moment. Second thing I just want to mention is test R65. So that's one of the tests in the National Genomic Test Directory, which you've just heard about. And this is taking us into the world of pharmacogenomics. So later on, Hayley's going to talk in much more detail about pharmacogenomics, but I just wanted to mention this test. So it's a test for a variant within one of the mitochondrial genes. And if you carry this variant, and about 1 in 500 people in the UK do carry it, then you are and if you carry this variant and you receive one of the aminoglycoside antibiotics, then it predisposes you to the risk of hearing loss and also balance problems. So therefore, if you've got this variant, we would avoid prescribing aminoglycosides to you. Now, if you think about people with cystic fibrosis, then as a group of patients, they're more likely to need these antibiotics for respiratory infections. So work is ongoing to ensure that all newborn babies diagnosed with CF also get the R65 test, so that we can try and avoid prescribing and these forms of antibiotics to those patients. So let's just try and move my slide on, bear with me. There we go. So just wanted to mention a few of the challenges that we have, therefore, in relation to CF and genomics. And it's already been mentioned today about the lack of diversity within the genomic data set. We certainly know about more variants in the white Northern European population than we do about variants in people from other parts of the world. So, you know, when we're thinking about research and research programs like the Generation Study, it's important for us to ensure that we capture a more diverse data set going forward. I've mentioned about the challenges with the new drugs in terms of access in this country, but also globally. And something that you probably don't think about much in prenatal diagnosis is you have to think about where that baby is going to be living. And over the years, I've counselled several couples who are planning to move to a different country, either temporarily or permanently, following the birth of their baby. And if that baby's got a specific condition, we have to think about whether these drugs or treatment are available in other countries. And we know that with these CFTR modulators, there's many countries that just can't afford them. So that's something that you do sometimes have to think about. Research is ongoing, of course, for that 10% of people with CF for whom the CFTR modulators are ineffective. And so, you know, we need to think about them in terms of our counselling and prenatal diagnosis going forward. And as more people with CF survive longer, then more of us are going to come into contact with these patients going forward. And it's just another example of why everyone needs to know about genomics and the impact on our healthcare services. This is particularly true in maternity. What we've found over the last few years is that more women with cystic fibrosis are able to conceive, possibly due to a combination of earlier diagnosis with our newborn screening program, but also with the new CFTR modulator drugs. In 2020, 56 women had a baby, or 56 women with CF had a baby, and in 2021 that was almost doubled to 103. So there's a challenge for maternity services to provide care for these women going forward. And I've also mentioned, you know, it's important that we find out who might be at risk of having a baby with cystic fibrosis early in pregnancy. So it's for midwives to ask those questions, to find those genomic red flags that we sometimes talk about, and refer people for specialist counselling as soon as possible in pregnancy. So I've nearly finished. I think this is my last slide. I covered an awful lot, I think, in just over 15 minutes, but there's a lot of information out there if you want more knowledge, and Genomics Education Programme website has lots of information and training courses, starting from what is a genome, what is a gene, right through to sort of information on master's level study. There's also G-Notes on there. G-Notes stands for Genomics Education, and Genomics G-Note stands for genomic notes, and it has information about all the tests that I've talked about today. There's a lot of information on there about cystic fibrosis as well. And if you're really interested in the drug therapy, because it's a fascinating story about how we've developed some of these new drugs for cystic fibrosis, then there is a webinar on that website that will in great detail talks about the development of these drugs, and I strongly recommend it if you're working in relation to cystic fibrosis in any aspect of care. Also got e-learning for health, so if you want to know more about the newborn screening program for CF, then do go and have a look at the newborn screening modules. And CF Trust has a lot of information on their website. Interestingly today, yesterday we noticed they had nothing about NIPD, or stated that NIPD was only available privately. So I've spoken to my lab colleagues, and we're going to get in touch with them to update their website. But there's a lot of information on there about the CFTR drugs and the availability. So I do recommend that you go and have a look at that. So I think that's all I wanted to say. I think the next slide has just got my
email address. So if you have got any questions about midwifery and genomics, do contact me. If you want anything, any other sessions, I can put those on for you. Thank you. Over to you, Phil.
So now we are going to go on to Felicity. Felicity is our regional Lynch syndrome nurse for the Central and South region, and Felicity is going to give us a talk on Lynch syndrome. So Felicity, when you're ready, go ahead.
Good afternoon, everyone. Thank you ever so much for inviting me. I'm going to have a bit of a whistle-stop tour this afternoon on the Lynch Transformation Project, but I will also sort of go into what Lynch syndrome is in a little bit more detail. If we can have the next slide, please. Okay, so what is Lynch syndrome? It's probably something that you've all heard, may have all heard of. Basically, it's Lynch syndrome is a germline pathogenic variant of one of our 4 DNA mismatch repair genes. And these effectively, a bit like what we were discussing earlier, help the body to protect it from certain cancers. And it's estimated that there's approximately 200,000 people with Lynch syndrome in the UK, and we know about 5% of those people. And we'll go into a little bit more detail why that's quite significant. So in terms of endometrial and colorectal cancer, which is what the project is mostly focusing on, 5% of those patients of those cancers are caused by a genetic alteration, and about 3% of those are either caused by Lynch syndrome. Can I have the next slide, please? So people with Lynch syndrome, as I've said, with the deficiencies in one of their mismatch repair genes, are at an increased risk of certain types of cancer. And this project is focusing primarily on colorectal and endometrial cancers. So for a general population, this slide is just sort of showing you how much of an increased risk they are at. So the general population, they are at a 5% risk of lifetime risk of colorectal cancer. If they have Lynch syndrome, that increases to an 80% lifetime risk of developing colorectal cancer. And similarly, with endometrial cancer, women or people with uteruses tend to be at a 2.8 lifetime risk of cancer. If we add Lynch syndrome into the mix, that does increase to a 60%. They are also at risk of other cancers, so things like ovarian, other cancers affecting the GI tract, bladder cancer, skin cancer, cancers affecting the nervous system as well. So there is a whole host of these things that they can be at risk of. Next slide. And the reason we want to know more really as to who is affected is Lynch syndrome is an inherited autosomal dominant gene, which basically means that if you've got parents and one of them is a carrier of Lynch syndrome, there's a 50% chance of you passing that to your children or you having Lynch syndrome. It doesn't skip generations. It continues down that road really. And so when we're looking at things like the NHS long-term plan, where they're looking at sort of prevention as much as cure, we want to know these patients who are affected by Lynch syndrome so that we can tailor treatments or surveillance to them. And what we'll look at is really how we're sort of discovering patients with Lynch syndrome in the next slide. So for the project, the NICE guidance back in 2017 basically said that we needed that all patients diagnosed with a colorectal or an endometrial cancer needs to have what's called an immunohistochemistry test. And that helps us to identify what genes are missing, if at all. So in colorectal, we see people who are younger, who have a younger diagnosis of colorectal cancer. Again, they tend to trigger something that might be genetic going on, so they would automatically get a genetic referral. So regarding which gene is affected will trigger either a direct referral to clinical genetics, or they will then have a further genetic test to have a look to see whether there's methylation present. And the reason for that is if, say, MLH1 methylation is present, then that the presence of that basically can switch off the MLH1 gene. So again, it may indicate to us that's more sporadic. So again, we're looking at sort of patients as well with a strong family history. So again, you know, we might talk about sort of other family members that have been affected by cancer as well. They would also be referred to clinical genetics. If we have, there is so obviously there's a colorectal side, there's an endometrial side as well. These can slightly differ from region to region, but generally this is what the national project is sort of handing out or advising really. So if we could have the next slide, please. So as I've sort of said, it affects your immediate patients, the patient that's diagnosed with a cancer. They can have personalised therapies, such as immunotherapy. So patients seem to respond very well to that sort of treatment. They can also have personalised advice and surveillance, and also some lifestyle recommendations, which we'll discuss a bit later on. And like I said, this isn't something that is just can affect that person. It can affect their families as well. So again, what we're looking at is predictive genetic testing. So for every one patient that we diagnose with Lynch syndrome, there's potentially 3 or 4 other family members that can be affected. So again, when we find out that those family members that are effective, we can obviously advise them on surveillance programs, and also they can have an effect on their family planning as well. We're very much linking with the genetics hub. So I'm local to Birmingham, so that's my main hub, and they obviously deal with a lot of the referrals. One of the things that we're trying to do as part of the project is to bring this germline or mainstreaming into everyday nursing practice or healthcare practice, so that we can sort of streamline and speed up that process for which a person is diagnosed with Lynch syndrome. So again, the genetic hubs are very much pinnacle to our project. They help to obviously test the family members. They help to offer with guidance as to surveillance recommendations. They obviously also can do additional testing for the tumour samples as well. And a bit from our earlier slides, there's 3 main results that a patient could potentially have. That's where we find pathogenic variant, which basically means that they have got Lynch syndrome. And then you sort of move into sort of the grayer areas. So the variant of unknown significance or the pathogenic variants not detected. And both of those basically don't rule out Lynch syndrome. So patients would still be, and their relatives would be recommended for surveillance. But again, just how their pathway follows differs slightly. If we can have the next slide, please. So in terms of the surveillance or lifestyle factors that can be recommended for patients, the main one really that most people may be aware of is the colonoscopy surveillance. So patients are recommended to have a colonoscopy every 2 years. And this can start from, depending on the genes, this can either start from the age of 25 or 35. And this can be particularly important for patients that you may meet that are in their 40s, that could have a deficient MMR and a Lynch syndrome diagnosis. The importance of, if they've got children and they're hitting the age of 25, for example, you want to try and get those patients into surveillance as soon as possible, really. And a colonoscopy isn't going to stop a cancer from forming, but the hope is, a bit like the bowel screening programme, is that we want to try and catch these cancers at an earlier time so that treatments are more effective. And obviously, again, a lot of cancers in the colon may start as a polyp, so again we can identify those patients earlier, and put them onto a regimented surveillance program. A lot of the colonoscopy surveillance now, or all of it, is managed by the National Bowel Cancer Screening Programme, which you may also be familiar with. This generally starts at 60. The age is getting lowered and it goes up to 45, but patients with Lynch syndrome who are at a younger age will obviously get put, and that surveillance will start sooner. Lifestyle factors, we can obviously recommend that there's studies going on at the moment to different things that people can do with Lynch syndrome that can help reduce their risk. So one thing again is aspirin. Aspirin does have an effect. A patient does need to be taking that aspirin for at least 2 years for it to have an effect. We're not quite sure on the dose at the moment. There is a study called the CAP3 study that's been headed up by Professor Sir John Burns, and that will hopefully give us the sort of right dose of aspirin to recommend to patients. They're also recommended to have a gastroscopy, so that's a camera test into their stomachs, and to have a look to see whether they're growing a bacteria called H. pylori, and if they are, then obviously eradication therapy that can also help reduce a risk of a gastric cancer. And also lifestyle modifications, like we said, cancers, whilst even though you've got a genetic condition, can also be enhanced by lifestyle factors, so weight, diet, drinking, smoking, but also through the research, through these CAP studies, they've also found things like green bananas can also help because of the starch that helps to protect the colon a little bit more as well. So it seems a bit of a random one, but the research is out there. So again, when we've got a patient that's been diagnosed with colorectal cancer, again, the treatments can be altered slightly. So they may be better off having an immunotherapy, which can help shrink the cancer a little bit more. So again, this is particular for, patients needing sort of low rectal surgery where they're having the rectum sort of completely removed. Again, there's discussions as to whether or not they have certain treatments first. So again, there is sort of guidance being developed on that, and also personalised oncological treatments as well. And for women who seem to get a rough end of the deal, unfortunately, if they are over the age of 40, they are recommended to have a total abdominal hysterectomy and bilateral saphengroophorectomy, which I probably absolutely butchered, sorry. Because at the moment, there isn't a satisfactory surveillance protocol for them. They felt that through research that having hysteroscopies and biopsies wasn't as an effective surveillance technique, mostly because obviously hysteroscopies, particularly outpatient ones done under a local anaesthetic, can be really painful. So they are recommended to have a hysterectomy. If you are younger than 40 and you're still in your sort of family planning age group, then again, we can look at certain fertility treatments, a bit like Karen was talking about earlier, where they can look at the embryos, and if they kind of take out the ones that are affected by Lynch syndrome. But again, what we tend to advise ladies, again, if they've still got their womb, it's very much about the symptoms. If they're ever worried, symptoms are the main sort of surveillance for this, really. Next slide. So a bit of a whistle-stop tour. I do apologise because I could probably rabbit on about but I wanted to kind of give you a bit of an overview. But I also just wanted to end on a bit of a positive story. So Mo Haig, he's written a book. He does come around to talk to us quite a bit. We've also got a patient that comes onto our talks who had a very similar story. So Mo was diagnosed with colorectal cancer at the age of 31. His family history gave him an access to genetic testing. So with his sort of everyday treatment, he was given 18 cycles of chemotherapy, had a major operation with a stoma and radiation for a balloon node in his neck. And basically his cancer kept spreading and sort of metastasizing, and was basically given a terminal diagnosis of about 6 months to live. And through his research, he was able to find out that immunotherapies was a treatment that could be offered to his group of his specific sort of group. He did some fundraising and managed to get access to this immunotherapy treatment, and basically with the immunotherapy it just melted all his tumours away, and is still alive now, and as I've put on the bottom, he's got a book as well. And they're very similar to other patients that have a very similar journey as well. We have a gentleman that comes again to do talks, and he was on a terminal diagnosis. He had liver and peritoneal mets, went and had some liver ablations done, and at the same time had some immunotherapy, and within a matter of weeks his tumours had all melted away. So there is a lot of research looking at whether we give immunotherapies to patients prophylactically as well, again just to sort of help reduce that cancer risk even further. So, next slide. So just like I said, lots of questions. I haven't put my email address on here, but I will put it in the chat. So happy to come and do any talks, to do training with teams on the pathways. There is a mainstreaming part to the other pathway, but I haven't really touched on that today, just because I wanted to give you a bit more of an overview. But if you have any questions, just pop them in the chat. Thank you.
Thanks very much, Flick. That was great. There is a comment in the chat from Lucille, who says when people come with Lynch come for surveillance, sometimes it's like a family reunion, brothers, sisters, cousins talking in the waiting room. And whilst for me, family reunions can be a bit of that or the other, it does seem it's obviously can be quite a scary time for many people as well. But thank you very much for your talk. And again, just for everybody as well, it's just to be aware that you've got Felicity. If you are having any questions about Lynch, Felicity is our regional nurse that will be able to guide you if you are working within colorectal or gynae. As we had Kate and Jess beforehand, if you have any questions relating to any tests or about genetic counselors, your counselors in your local trust, your genetic counselors and the lab, they are all very good resources to get your questions if you have any queries for any patients you are seeing. But going forward now, we have got our next speaker coming up, and our next speaker is Michael Carter, and Michael is the CNS who is based at Wessex Clinical Genomics Service, and is the CNS leading our team of genomic practitioners. So Michael, if you're ready, you are going to talk to us about the nurse's perspective of whole genome sequencing. And are you moving your slides, or is Donna moving your slides for you?
I'm happy either way. Donna can do it if she's got access, if she can control it, that's fine. Lovely. So yeah, as Phil said, my name's Mike. I'm a Genomics CNS based down here in Southampton. I'm just going to talk to you a little bit about how, as sort of give a bit of the nursing perspective on whole genome sequencing. But the next slide. So I know you've already had a talk from Kate in the lab about genomic testing techniques, so I won't go into too much detail, but I'll just quickly recap on WGS, what it is we need for WGS to go ahead. and then talk about how the nursing role sort of fits into that. So why is it useful to have nurses in that WGS pipeline? Talk to you about the consenting. Again, I know Kate's talked to you about some of the forms and things, but I'll talk to you a bit more about that discussion that we have with the patients. And then I was going to finish up by talking a little bit about certainly how we work down here in Wessex with some of our CNSs who are currently already doing WGS in mainstream, sort of what they get up to. And also thought it'd be useful and interesting, hopefully, to tell you a bit about what I get up to as well, because I think we talk a lot about mainstreaming, but actually I think it might be nice for some of you guys in mainstream to actually see what a bit of what a nurse might do in clinical genetics as well. So I'll talk to you a little bit about as well, and then I'll speculate a little bit about the future. Next slide, please. So hopefully, as you've already heard, so whole genome sequencing is a pretty large genetic test. As the name suggests, sequences the entire genome of a patient. And there are two main arms to this. So there's the rare disease side and the cancer side. Rare disease, as you can see in that diagram, is often done as families. So a patient with samples from each parent called trio testing. Although it can be done as duo if we only have one parent, or singleton if we don't have any parents. And the advantage of doing that as a family is that you can see what's been inherited from each parent and what might be brand new or de novo in the patient, which is why that is useful. And then with cancer, a little bit different. You can compare a patient's germline DNA, so DNA from their healthy cells, against DNA from their tumour cells, and compare and contrast, so that you can then see what's sort of happened genetically in the tumour and sort of try and get a bit more information about their tumour and whether that may have any influence on any treatment plans. But it will also look for things like germline variants as well, so genetic changes in their healthy cell DNA that may have perhaps predisposed them to cancer as well. So there's obviously family implications in that. So WGS will sequence essentially almost all known genes across the whole genome. So that's about 3 billion letters of code. And that includes both that 1.5% of your genome that codes for proteins, but also that 98.5% of your genome which doesn't. So it's a huge amount of data. And then the lab will use panels based on the phenotype of the patient, so how they're clinically presenting, to choose which genes to look at. So you might ask, why do they sequence the whole thing if they're only going to look at a small section of it? And one of the key advantages of this test is because we have all that DNA sequenced, if we don't get an answer straight away, you've got all that data there, so you can go back and reanalyze in the future as and when we have more up-to-date knowledge through research, or if the patient has new features showing, then you potentially go and reanalyze some of that data and get answers down the line. So we've got the option for reanalysis, and also we are able to offer patients having whole genome sequencing the option for their data, their genomic data, to become part of the National Genomic Research Library. And for some patients who don't get an answer through the NHS side of the testing, do actually get answers down the line from novel findings in research. So that's something we are able to offer patients as well. Next slide, please. So for WGS to go ahead, there's a few things, and if you speak to anyone who does WGS already, I'm sure they can tell you that it is a time-consuming, lots of administrative tasks, and all around quite clunky process, because we need lots of different things to get it going. So we need a test order form, which I think Kate's already talked to you about, which gives just the clinical information about the patient. We need record discussion forms, that's our consent forms, and we need one for every member who's having the test. So if you've got doing a trio, then we need one for every member of the trio. And then obviously we need the appropriate samples. So for rare disease, we usually use blood samples. And then for cancer, obviously you need a tumour sample and then a germline sample. And that might vary depending on what type of cancer it is. So that's WGS in a nutshell. So if I can have the next slide, we think a bit more about how the nursing side fits into this. So it's part of the NMC proficiencies for registered nurses to know about genomics. I'm not going to read these statements out, but these are part of that document. And this was published in 2018, so this is 5 years ago now. But what we do know is that nurses are still kind of falling quite behind with that genomics education. And I've just referenced this study by Thomas et al., who did a big literature review earlier this year, looking at all the papers published on nursing and genomics education from the last decade. And so some of the key themes throughout that was that nurses were sort of consistently reported to feel like they don't have the knowledge to talk to patients confidently about genomic testing, or they don't understand how genomics would apply to their specialities. So although genomics has been on the board for nurses for a little while, it's still taking a while to get in there a little bit. So if I can have the next slide, please, and I'd like to tell you a bit about why I think it's useful having nurses involved, particularly in whole genome sequencing. And obviously this, can obviously extend to midwives and other healthcare professionals as well. I'm just talking from my experience as a nurse, but as nurses and healthcare professionals, we're the ones who know the patients probably more than any other. We're often the first point of call for those patients because of that, particularly in those specialist nurse roles. And that puts you in a really good position, because we know when it's appropriate to have discussions with patients. We know what the patients are going through at that time, and for parents as well, if it's pediatrics. Certainly I wouldn't want to be calling up a parent of a pediatric oncology patient out of the blue to talk to them about, have a 20-minute discussion about genetic testing, when I don't know where they are in their treatment, what's going on with them. So I think it's really helpful for a CNS who knows those patients to have those conversations. But by doing that, the nurse actually becomes more informed of that patient's care. They're more involved in that patient's care. And then this feeds back into this cycle of, actually, we now know our patient even better. So it's a really useful thing to be involved in, because it's beneficial for both the nurse and it's beneficial for the patient as well. So if I could have the next slide. Lovely. So one of the key areas which nurses have been consistently involved in with WGS down here in Wessex, certainly, is having this discussion around the consenting for whole genome sequencing testing. I'm not going to go through the whole of this form with you, but I just wanted to sort of give you a very brief flavour of what we do discuss. This normally take about a sort of 15-20 minute conversation, because we need to make sure patients understand what it is they're agreeing to, and give them the opportunity to ask questions, because this is quite a big test to have, and there's a lot of things we can find out. And as we know with genetic testing, once we find out, you can't unfind out. So it's really important patients understand what they're saying yes to. And to be quite honest, for a clinician who has a 20-minute appointment with a patient, you can't practically have a good in-depth discussion about this in a 20-minute appointment. So I think this is where, again, having that CNS involvement has been really useful. But so when I speak to patients, what things do I kind of make sure they know? So first of all, I want to make sure they know what whole genome sequencing is. I don't get into massive depth. It's a complex test. I generally try to avoid too many analogies. Analogies I find can be helpful, but also can sometimes take away from what you're trying to get across as well. So I generally try to avoid them, but may occasionally use them. So I want the patient to know in a nutshell, what the test is. And then what I want to know is what could come back from this test. Now with WGS, it's not a case of we find something we don't. There's sort of 4 potential results we could get from a WGS test. We might find a genetic cause for a condition. Obviously, that'll be fed back to the clinician and fed back to the patient. and that's obviously what we ideally want. Of course, we need to manage expectations, because although this is a very comprehensive test, we just might not find anything. And that is a very common result as well. But equally, because we have that opportunity for reanalysis down the line, it might be we can still get answers in sort of years to come when we have more knowledge. It's important that we make patients aware that some results may be uncertain. So these are the variants of unknown significance, which I know Jess talked a little bit about earlier, where we're not sure whether the genetic change seen is perhaps part of normal genetic variation, or whether it's whether it's potentially a cause for a condition. We just don't know at this moment in time, and it might be in the future we can relook at that variant and get an answer down the line. But these are uncertain results that aren't going to be actioned. But also we know from research that's been done that VUSs can also be a point of frustration for families, particularly parents of paediatrics, because there's been a gene named on a result on a report, but apparently it's not at this moment relevant in time, and that can be quite a frustrating thing for patients. So we make patients aware this can happen. And lastly, the fourth type of result we need to make patients aware of is that we might get incidental findings. So that's findings unrelated to why a patient's having this test, but it might have wider family implications. So, for example, finding a genetic change that might show that they're predisposed to certain types of cancer, for example. It's rare that these are found because the scientists aren't directly looking for them, but at the same time, it's not an impossibility. So it's something we do need to make families aware of. Other things that we do make families aware of as well, I won't talk about data and storage because I know you have a talk coming up on data ahead, but other things that are important to mention, particularly for trio testing, is this test will check that both parents are the biological parents. It may seem like a stupid thing to say, but it's definitely better to have that conversation at the start than get a result from the lab in a year's time saying that there's a biological mismatch. Stranger things have happened. So I won't say any more about the record discussion. Oh, and also we do talk about insurance as well, and offer the patients the opportunity for the research library. But in the interests of time, I will move on. So if I can have the next slide. So last couple of minutes, I just want to tell you a little about what some of the nurses down here get up to in both mainstream and also in clinical genetics. So we have various CNSs in completely different specialities across our region who are doing WGS. So you've got some over in peds epilepsy, we've got some in oncology and immunology as well, who are all organising WGS because they're in a really good position. They're part of that MDT, that wider MDT, so they can help identify suitable patients. They're then, as I said earlier, they're knowing when it's appropriate to speak to those patients, having those consent discussions, organising all that paperwork, getting those samples organised, and then they're there as a point of contact for that patient as well. And also for the lab, a point of contact for the lab to come back to if there's any queries down the line. So that's how it's working with a lot of nurses in mainstream down here, and that works really, really well. And if I say so, arguably it's slightly better than where we would try and get more medical teams to do WGS. But that's because, as we know, a lot of medical teams, they rotate, particularly the junior staff, whereas CNSs are likely to be in their roles for much longer periods of time. So that's, I think, one of the reasons why it sort of has worked as quite a good model down here. And then if I can have the next slide. And I thought it'd be good for you to see a bit of what I do as a genomics CNS, because like I say, we talk a lot about mainstream, but we don't really talk about actually what happens in clinical genetics. And I thought it might be useful to see what a nurse does in a role, which there aren't many of us. So in my role, so I still have a lot of discussions around families and patients about consenting for whole genome sequencing. And a lot of clinicians will ask our team to organise that, both from clinical genetics and from mainstream areas. And as part of that, I manage the Genomes team, which is a team of genomic practitioners who go through these consent discussions with families, but they're not registered health professionals. They're usually science graduates. Many of them go on to become trainee genetic counsellors, and they have an interest in genetics, but they're not registered health professionals. So actually you do need someone with clinical experience sort of to manage those teams. So that's what I do. And as part of that role, I also sort of have some more project management-y type responsibilities to help push WGS into new areas, and also support those teams who are already doing WGS as well to make sure they're keeping going, offer training to new team members and things like that. So quite a lot I kind of get up to. So last slide, well, second to last slide. So looking forward, and this is purely me speculating, looking forward, how nursing in WGS is going to look. As we know, genomics is getting bigger and bigger. There's going to be increasing opportunities for nurses to get involved, and for midwives and other health professionals as well. And personally, I think what we're going to see more of is not necessarily lots more of people like me doing a full-time genomics role, but actually we're going to see more and more of genomics being incorporated, particularly into CNS type roles. I think that's really important and definitely something to start considering. Thing is, WGS, as I said, it's time consuming. So actually we need to start thinking about having time put aside to do this rather than adding it on top of already full job descriptions as an extra, because you do need that time to be able to do this well. But like I say, that's purely me being a bit speculative. So next slide, I've just popped some resources there for you, some great links there. The e-learning for health do some great genomics teaching modules. I expect someone might have already mentioned this already today. And they'll do some great genomics modules, and also on whole genome sequencing as well, and some other things for you there. So that is me. I don't think are we doing questions? Or are we just doing questions on the chat?
We'll do questions later if you are happy to stay on till the end, Michael. Otherwise, if we can come back to you if there's any questions as well, that if you don't have time to stay. But thank you so much for that talk. I think it is really important, and thinking about nursing specifically, that this is something that is coming. It's going to come in. It will be an even bigger part of our roles going forward, I think, but thinking about the NMC and our standards of proficiency, our education that we're getting in our training is not great at the moment, but that will change. But and there are going to be very many more roles I see in the future for nurses and genomics. So thank you very much. We've got our next speaker now, and we have got Professor Sarah Ennis, who is a professor of genomics here in Southampton at the University of Southampton. And Sarah, Professor, is going to speak about, oh my goodness, I'm sorry, is going to speak about genomics data ethics and research. So Sarah, if you're ready, I will hand over to you. Sorry, Donna, I think.
Can you see and hear me and see my slides?
We can see you. Sorry, Donna, I think we are sharing your screen here at the moment as well.
Oh, I'll stop sharing. Sorry, bear with me a second.
Okay, I'll
there you go.
Can you see my slides now?
Yes, we
thanks,
can.
Sarah.
Lovely. All right.
Sorry,
okay.
Donna, it's back.
I'm not quite sure what you can see. I know what
no, Donna.
We just need Donna to stop sharing his screen. That's it.
It's not stopping.
Well, Donna, if you prefer, I can stop. Yeah.
Go ahead, Sarah.
Okay, so you can see my slides here right now.
Yes.
Yeah, lovely. Okay, thanks very much for having me, Phil and Donna. So for those of you who don't know me, I'm Sarah Ennis. I am a research director in the Central and South Genomic Medicine Service, but I'm also a professor of genomics at the University of Southampton. So I've been working in analysing genomic data in derived from patient samples for quite a few years, since the last century, in fact, since before the human genome was mapped. So when we knew very, very little, but we were starting to diagnose some rare diseases back then and do some association studies with complex diseases and to try and track the genetic causes of cancer. But of course, things have come a very, very long way in the last 20 years with the mapping of the Human Genome Project. And so I continue research in that area, but I also have a sort of research-facing role in the Genomic Medicine Service. So I'm going to talk a little bit about data ethics and research today with respect to the Genomic Medicine Service data. So great. So I'm sure you may even have already seen this slide today, but just to talk very briefly about the steps in the clinical application of genomics. So it all starts with identifying the actual patient and or their families, be that for cancer or for rare disease. And then, as Michael so very nicely detailed in the last talk, the conversation around consent in that record of discussion. So that's clearly a very, very important role, and one that nurses play a huge role in. And that's critical. It's both for the Genomic Medicine Service, it was for Genomics England, it is in any of the research we do, in having that patient-facing individual that is trusted by the patients, that they're free to ask all these questions and air their concerns with respect to genomic testing. So you have that discussion and you record consent. And of course, within that, there is the consent for research. It's followed by the sample collection. And then once the sample is collected, it's typically sent off to the laboratory for whole genome sequencing. It gets packaged up, the test order data from the test order form and data from the record of discussion, or the fact that that's been achieved, is all recorded. And the samples are then sent to this huge sequencing service or facility up in Cambridge. And from that DNA sample that's extracted from the patient or the mum, dad and patient, you generate these sequencing data from these massive sequencing machines. And those data are uploaded onto a cloud, and they undergo something called alignment, where they're aligned and mapped back to the human genome sequence for each individual. And then you basically do this compare and contrast, this spot the difference. Of course, we're talking about 3 billion base pairs here. So computers actually do the spot the difference, and they call the variants that are different in that patient and perhaps that patient's parents, or that patient and their tumor. They call those differences and generate what's called a variant call file. So the positions in the genome where the individual's genome is different to the human genome reference sequence. And that variant call file is the basis upon which both the clinical report is written and upon which any downstream research is conducted. So that involves this sequence alignment, annotation, and sometimes validation. Of course, it's very important to make sure that, as Michael alluded to, that the sample is the sample you think it is, both with respect to the relatedness to parents or other family members, and to look at the ethnicity, to make sure the gender lines up. So these sort of quality control checks that you do. And then when those data are made available, an alert is sent out to the referring Genomic Medicine Service to say the data have been aligned and they're sitting there in the research portal, and the clinical scientists can log in and they can then assess those data against whatever panel has been recommended for that patient on the test order form. So if, for example, it's a patient with epilepsy, there may be a specific epilepsy panel that's been referred to, and the clinical scientist will now interrogate those variant data for mutations in genes that are known to cause, for example, epilepsy. And if they find variants that are known to be causal, those are the things that will make it onto the PDF form that are returned back to the clinician. So they're reported back to the consultant first, and then of course back to the patient and family, and perhaps to wider family members if they want to conduct downstream screening. And of course that is the basis of any targeted treatment or precision treatment, or treatment that's guided by understanding what is the genetic basis of that disease. And of course, there may be counselling involved, depending on what the diagnosis turns out to be at that point. So that's the rough pathway. Of course, consent it's a very important issue, and it's important that the patients, before they give their consent, that they have been provided sufficient and appropriate information. Of course, the nursing teams and the patient-facing teams are critical in conveying that information and explaining it where people have concerns. So the individual has to be competent, able to retain, process, and understand the implications, and it has to be voluntarily given. And it's important here just to know that consent is just one of the bases that allows us to share data. And there's actually 6 lawful bases, and the other 5 include contract, legal obligation, vital interests, public task, or legitimate interests. Now, I'm not going to go into those today, but it's just good to understand that consent is one of the bases for sharing data. And sometimes it may be in the future that some of these other bases are also looked at. The record of discussion, Michael's gone through this really nicely already. From the perspective of data, of course, individuals are allowing for their biological sample, their DNA sample to be taken. And within that DNA code is the data about their molecular susceptibility to disease. They're, of course, allowing for the data to be stored. It's very expensive to generate and to process these data. So it's important that we store the data, much like any other medical test record. And that they allow for their health records to be interrogated, because of course it's entirely possible that, for example, a 2-year-old child could be referred with kidney failure or protein or blood in their kidneys. But of course, and that may be correct on the date that that test order form is filled in, but patients, especially children's diagnoses and symptoms change over time. And for adults with cancer, their response to drugs may not be known at the time that their original biopsy sample is being sent in. So it is important that individuals consent to their health records being looked at in the future to understand, well, okay, you presented with kidney failure, but in actual fact, a few months later, they started to develop hearing problems or deafness. That could be really, really important information when trying to interpret and analyze genomic data. Or, for example, that you were diagnosed with a breast cancer, but to understand that it was a breast cancer that did not respond to treatment X, Y, or Z, that can be really, really important for us being able to understand and inform and help support decisions for other patients in the future, to understand that cancers caused by one set of variants may or may not respond to any particular drug. So that enriches the data for everybody's benefit. And that's why I would come back to this point here, that there can be other interests, so a public interest in using these very expensive data to actually not just inform for the patient that you're looking at today, but perhaps for other individuals in the future or next generations of those from those families. So this is the pathway as it looks like for research. So patient contact points, the patient chooses to contribute to the National Genomic Research Library. So that really is the Genomics England research environment at the moment. There may be changes to that consent over time, and there can be obstacles or administration involved if you consent a patient or their parents consent on their behalf when the patient is maybe 5 or 6 years old. Of course, that individual has a right to reconsent themselves once they achieve competency to do so. So occasionally, some of the data that's available to us for research, we may be screened off from seeing that until a second consent has been obtained after that individual reaches, for example, the age of 16. There are very, very strict security arrangements when the data are loaded into this research library or this research environment. So anybody who's choosing to access the data must be registered with Genomics England. They have to be verified as coming from a reputable institution, perhaps either for academic research or they're an industrial collaborator. And so their origins, their affiliations must be verified before their accounts are proven. And at that point, then they get multi-factor authentication and very secure access into this research environment to enable them to look at the data. And the data that researchers see at that point is, of course, linked anonymized, which means that somebody somewhere, if you find out, oh, okay, this patient had epilepsy and we think we found the cause of their epilepsy, there has to be a way to get that information back to the patient. But the researchers never see that. They never see patient identifiable data, and that piece of information is sent back through a diagnostic form back through the Genomics England administration, and they filter it through to the diagnostic discovery panel, who filter it through to the consultants and back to the patients that way. So it's a very, very secure environment. Any data that might come out down on this pathway, so the researcher applies for online access, they get verified whether they're from industry or from academia. There's a research oversight committee, and anything that's to come out of the Genomics England environment has to come through this red airlock. And what might come out, for example, if you do discover that, oh gosh, this gene that we never understood the function of before, if you have mutations in it, it can cause epilepsy. Well, it may be that that could be the case for other patients, either in Genomics England or in the rest of the UK or in other places around the world. So it's very important that you convey that to a wider research community. And in order to do that, you must put details of the publication that you want to publish, of the data, and specifically the genomic data and the phenotype data, so that's the clinical characteristics of the patient. You must detail those to the airlock committee. It must be reviewed by them. The integrity of the patient anonymity is looked at very closely by the airlock committee, and then they either approve or disapprove that release of data through the airlock for downstream publication. This is how the Genomics England new research environment is going to look going forward. There's been quite a restructure and a change recently, so it's got these sort of various aspects of its structure, whereby it's looking at diversity of approaches, it's changing its governance, it's public and patient engagement, it's got proactive support, and the community and collaborations. This is really where the research happens. And the research communities have been restructured. They used to be quite organ-based, but now they're a little bit more pan clinical speciality. And you can see that you have cancer and molecular oncology, discovery and interpretation, predisposition and screening, therapeutic innovation and trials, bioinformatics and machine learning, genotype, phenotype, implementation and data enhancement, and population genomics. So these are the 8 new communities under which every single researcher will belong to one or more of these communities and conduct their research under that umbrella and have a network and a community that they can work with there. At the moment in Genomics England, we have all the data from the 100,000 Genomes Project. So that's about 106,270 genomes across 90,000 participants. The vast majority of those were rare disease patients and their parents. So about just over 70,000 of them were from rare disease patients. So often that meant 3 genomes, because you had typically mum, dad, and a child. There are occasional families where there might be 4 members, or some exceptions made where there's only 2, but the majority of them are trios, mum, dad, and child. And then of course there are about 17,000 cancer genomes. So that means about 8,500 individuals with cancer, and of course a DNA sample from their tumor as well. At last count, there were at about 6,000 genomes from the Genomic Medicine Service. So this is where people like Michael in the last speaker, he's consulting or he's speaking to people on a day-to-day basis, getting their record of discussion, they're getting their genome sequenced, and that's happening all over the country. So those new genomes are being loaded into the Genomics England research environment now. There's only about 6,000 at the moment, but it's expected, I think, over the next somewhere between 6 and 18 months, I expect the number coming from the Genomic Medicine Service should probably outnumber those from the 100,000 Genomes Project. And of course, we have other projects like the Generations Project starting off as well. And there is plans for additional transcriptomic and proteomic data that will become available. And I'll talk a little bit about those in a second. So sequencing data, the data that are generated are largely what we've talked about so far are these whole genome sequencing data, where for each individual, you're sequencing the 3 billion base pairs they got from their mum and the 3 billion base pairs they got from their dad, and that's the whole genome. And they're doing that by a sequencing technology largely run by the Illumina sequencing company, whereby they chop the genome into these short bits and sequence those short bits and then reassemble them all and align them and call all the variants. But there are other approaches that are still being used in the genomic medicine service. Some of them is whole exome sequencing. So exomes is not sequencing the entire genome. It's just sequencing the 1 to 2% of the genome that has the DNA base pair code that makes proteins. So it's a very specific targeted method of sequencing. You're looking at all the genes, but only the bits of the genes that code for protein. And so it's a vast reduction in the amount of data you generate, which can be a good thing. These data files can be enormous, very expensive to store, very hard to analyze. So whole exomes is a sort of smaller, more condensed type of data you can get, but it does miss some important things that you would only capture with whole genome. And then even more targeted than that is something whereby you just go in and you target maybe 5 genes or 50 genes or maybe 500 cancer genes, and you just look at the coding region of those genes. And again, this is smaller amounts of data, faster to analyze, and these sort of panels are used quite frequently in cancers and to very good effect. So instead of doing the whole genome, you look at the set of cancer genes that are known to be implicated in those cancers. And then there's this other thing that's being done increasingly at the moment where it's not DNA you're sequencing at all. So DNA is kind of like the recipe book that resides in the nucleus of the cell. RNA is the bit of molecules whereby it goes in, opens up the DNA, reads the recipe, takes a copy of it, and takes that recipe back out outside the nucleus and turns it into a protein. And that's called messenger RNA. And if you sequence that, that's called sequencing the transcripts or the transcriptome. So you're sequencing not DNA, you're sequencing RNA. And that is being done as part of some of the transformation projects and now the new genomic networks of excellence that exist in the genomic medicine service. And so it's something that I expect we'll probably see a little bit more of in the future, and that we may need to explain to patients what we're doing there as well, not least because we're going to use those data in research and to help us interpret the variants that we see in genomes or exomes. We actually can use the RNA to help us interpret as to what the functional consequences of those genetic variants might be. Another thing that's changing at the moment, I've mentioned how the genome tends to be chopped up into these short little sequences, sequenced and then reassembled into the genome. There are advantages to doing something, to chopping the DNA into not short sequences, into actually having it in quite long sequences. And that's called long read sequencing. There are some other companies that are providing long read sequencing data. Oxford Nanopore Technologies, Pacific Biosciences, and Illumina themselves have an offering that purports to provide long read data. But these are additional data that we may also be generating on patients in the future. Long read data can be very, very useful for looking at changes in the genome over tricky areas where there's structural variation. In cancers, we can have a whole section of DNA gets chopped out and turned upside down and reinserted, or maybe reinserted into the wrong place. And to try and understand those sort of structural changes in the genome, long read data is much, much more sensitive to pick those up. Phasing, that means working out which variants are from mum and which variants are from dad, rather than having a soup of all the variants you inherited from mum and dad in one big long list. You actually separate them out into those from mum and those from dad. And that can be really, really important if we're trying to understand recessive diseases in particular. So again, I think that's going to be a driver towards generating long read data in the future. Either way, we generate huge amounts of data, and this is one of the big problems with genomic sequencing and long read data. We generate enormous files. So we had a trio of whole genome sequencing data transferred into our research environment. It didn't come from the Genomic Medicine Service, it came from elsewhere, but it's about 400 gigabytes of data. And when you think about, you know, one high definition nice photograph that you take this Christmas could be a couple of megabases. This is 400 gigabytes just for one trio of data, of whole genome sequence data. And a single individual's long read whole genome sequence data might even be double that. And that excludes some of the really important files. So this is one of the issues that we have with genomic sequencing data and some of the problems. But we are going to try and overcome them, because there is all these potential benefits. And of course, by pushing the data and using the data in research, we hope to get better at identifying and making diagnoses earlier, making those more precise diagnoses, and being able to, once you identify, for example, exactly which transporter is malfunctioning in patient A's epilepsy as opposed to patient B's epilepsy, well, then you can target that particular transporter that's dysfunctional in patient A, and you can target the different problem that's dysfunctional in patient B. So you get the correct drugs to the correct individuals in a timely fashion. And you can also help avoid adverse reactions. So as well as identifying reasons why people might get sick or be ill or have syndromes or have cancer, we can also look at how the liver processes drugs, and we can try and identify if there are changes in any of those enzymes in the liver that might mean somebody doesn't process a drug very well, or might experience an adverse reaction. So we can avoid those. We can avoid causing harms by looking at different places in the same genomic data test, if you like. And therefore, and we can also look at the eligibility for patients for clinical trials, especially if they've been refractory to typical treatments. It can be very good to identify, well, these are the patients that didn't respond to the first line treatments. There are some new treatments coming on board that perhaps we might like to try. And we heard about some of those really good examples in the talk on Lynch syndrome. So I will leave it there. I believe I'm over time. So I'm not sure if I'm going to take questions now. Back to you, Donna.
Thank you very much, Sarah.
Or Phil.
That was a really great comprehensive view of data and sequencing. Now, I know you said you've got a meeting, so
I have 6 minutes.
You have 6 minutes. So I wonder if Hayley, Dr. Hayley, is okay if we can just go to, there's 2 questions that have been put into the chat. So the first one is, is, are genomic medicine students allowed to apply to access data for their research projects?
Genomic medicine students, yes, they are, but they would have to be affiliated with a university, and they would need to be affiliated with a project, or possibly start their own project. So there is a very clear and transparent way. One, you need to be registered with the university and a bona fide student there. You will probably need a supervisor, and your supervisor would need to be registered, and it would be possible to put your project and submit your project. Time is going to be of the essence, because sometimes these wheels turn a little bit slower than you might hope. So I would say get onto the website today, explain that you're a student, and depending on the complexities of your project, it might be possible to get approved really, really quickly and get in there and look at the data. But that is absolutely, students are welcome, but you need to be registered, and therefore you need to be verified as being either with an approved company or an approved university. And I would expect having some a proper supervisor on the project and having them registered would be useful as well.
Thank you. And the next question is from Peter Newton to say, NHSE is implementing a research data warehousing function called SDE regionally on PID. Okay.
So NHS England have started these sub-national secure data environments. It's a massive mouthful, but it basically means it's going to be these data lakes that hold NHS clinical data in a secure, harmonized, standardized format. And they're being rolled out. There's a number of them across the country. There's one in Birmingham, there's one in Oxford, and there's one in Wessex. They're not all doing exactly the same thing, but they will all be expected to be able to talk to and from the Genomics England and the National Genomics Research Environment, or the Unified Genomic Record, as that might evolve over coming years. So these massive data repositories, it's absolutely critical that they're able to talk to one another, because it's one thing, you must have the longitudinal clinical data alongside the genomic data. And so it is absolutely important that these things talk to one another. To be absolutely honest, it is not abundantly clear to me the strategy on how that's going to be achieved as yet. But I do know that there are some people on the ground in at least some of those secure data environments that are doing their best to try and make sure that those data can link up. But there's a little bit of siloing, unfortunately, I think, at the moment, but the value in merging high caliber clinical data and these genomic data is too high not for that to happen. But I can't quite agree that perhaps everybody's singing to the same hymn sheet right now. I'm not sure the hymn sheets are all written.
Thank you very much. I hope that's answered your question, Peter. Thank you so much, Sarah, for your time.
Not at all.
It's really been great.
Yeah, thanks, Bill. And sorry, Hayley, that I had to take those, and see you all soon. Bye-bye.
Thank you. Bye. So on to our next speaker then. We've got Dr. Hayley Wickens, who's our consultant pharmacist for the Genomics Medicine Service Alliance. And Hayley is going to talk to us about pharmacogenomics. Very exciting. So on to you, Hayley.
Thanks, Phil, and thanks for the invitation to talk today. I know some people may need to drop off, but I believe this is being recorded so you can access it later. So so my name's Hayley. I am a pharmacist. I specialise in genomics. I work for Central and South. And what we're going to talk about today is how we can apply genomics to help patients get the most out of their medicines. Now, prescribing medicines is pretty much the most common intervention in the NHS. So it's really important that we get this right, and we often don't. But we'll talk about how this can make things better. So we'll talk about what is pharmacogenomics. We'll talk about the use of PGX, that's an abbreviation for pharmacogenomics, in treatment stratification. I'll explain what I mean by that in a minute. We'll go through some examples of pharmacogenomic testing, including those that are commissioned and some that might be coming in the future. So we'll move on to what's coming next and then finish up with where to find out more. So as you've heard repeatedly this morning, and it's been some fascinating talks, but I find this absolutely mind-blowing. If you laid out your DNA from one cell end to end, you have 3 billion bases. That's a lot of letters. So remember, A, T, C, and G, your letters in your DNA code. But you can change a single one of those and have a profound effect on the patient's condition. For instance, sickle cell disease is a single base change. So is achondroplasia. But it means that some tiny changes can have profound impact. With medicines, we've now finding that we can look at those changes, sometimes 1 or 2 bases, sometimes bigger patches of changes can alter the way you handle medicines. And that's what we're talking about when we say pharmacogenomics. So this is the Oxford English Dictionary definition of pharmacogenomics: the branch of genetics concerned with the way in which an individual's genetic attributes affect their likely response to medicines. So this is a sort of new era for us, because historically what we've tended to do is put the drugs into the patients and then see what happens. And we do have prescribing guidelines, where we can slightly tailor treatment. But typically, if you have a patient group that's got the same clinical diagnosis, you give them the same prescription, there are 1 of 4 possible outcomes from this. So ideally, you'd want everybody who's given the medicine to treat their condition to come out in group 3 on the bottom right here, where the drug is not toxic, so they don't get a side effect, and the drug works, so it's beneficial. Yay, that's great. Unfortunately, we know that's not the case. Adverse drug reaction-related admissions to hospitals in the UK run anywhere between 6 and 15% of admissions are down to ADRs. Depends on which paper you read, but it means that this has got a huge cost to people, humans, but it also has a huge financial cost to the NHS in the order of billions of pounds. So this is what happens when things go wrong. People can have the drug and they go into this group here where the drug is toxic and not beneficial. So they're getting an ADR and the drug doesn't work either. So that's the worst possible outcome. But we know that happens. That causes those admissions. In group 1 at the top left here, you've got where the drug is toxic. So you get an ADR, but it does work. It's beneficial. It has the therapeutic effect. So then you're into sort of risk management decisions about whether you use that drug in that patient. And then group 4 down here, the drug is not toxic, so it doesn't give you a side effect, but also it doesn't work therapeutically, which obviously is a waste of everybody's time and money because the drug's not doing anything. And also the patient is potentially delayed in getting to an effective therapy. So what we're trying to do using the science of genomics and pharmacogenomics is to predict where people are going to end up when we give them a particular medicine. And you think, well, that might sound very Star Trek and very new, but actually it's not. One really famous example of this is abacavir, which is an antiretroviral drug used in HIV, very, very effective at killing the virus. So we automatically know that it's beneficial for everybody. So people go into group 1 or group 3. So the drug is beneficial. It's either toxic or non-toxic. But what we can do, and we have been doing this since the early 2000s, if you're going to start abacavir in someone, we'll go and look at your gene sequence or your genotype in your major histocompatibility complex. So this is your human leukocyte antigen gene. So we can go and look at the sequence there. And if you have a particular sequence that we know of, we know that you're at high risk of getting a really severe reaction to abacavir, which causes like severe mucosal problems. And it's severe enough to get you admitted to hospital. But we know we can pick that up. So we do a genetic screen, check if you've got the variant. If you have, you don't get abacavir, you get a different antiretroviral. If you don't have the variant, then you get abacavir. So we've been doing this for 20 years. It's not that new. And you might think, oh, okay, well, you know, we've heard about a few cases of use today, but it's quite niche, really, HIV or cancer. It's not all of our patients. Well, I'm here to tell you, actually, it's a lot more than you think. So this was a nice piece of research done by a PhD pharmacist called Ezra Yusuf at UEA, who must have finished by now. I keep quoting her paper. But she looked back at general practice prescribing records in the UK, and she looked at just the top 5 pharmacogenes, so genes that are involved in drug metabolism or adverse events. So typically these are liver enzymes and other ones that are involved in drug targeting or metabolism. She looked at those 5 top pharmacogenes and all of the prescriptions. And she worked out that about 1 in 5 new prescriptions would require an increased dose, a decreased dose, or avoided altogether because of a risk of adverse event. And that's 1 in 5 new prescriptions. So this is not just sort of specialist medicine. This is pretty much across the board. 99% of us in this meeting today, if you did the numbers, will have at least one variant in our genes that affects drug metabolism. Obviously, if you don't get given the drug, it won't make any difference, but it shows you how widespread this is. Okay, now this is a potential application, and it's for a very, very common drug, codeine. So not only do we use this every day in hospitals, it's bought over the counter, it's used in community practice, it's used all over the place, commonly prescribed analgesic. But codeine is a prodrug, doesn't work unless it's activated by a liver enzyme called cytochrome 2d6. And we know that the cytochrome 2d6 gene that makes that enzyme has many, many possible variants, which can affect how well the resultant enzyme will convert codeine into the active drug, which is morphine. So you can have people who are slow metabolizers, who won't get much pain relief because they don't convert the prodrug codeine into the active morphine. We can get people who are ultra rapid metabolizers at the other end of the scale. So they convert that codeine into that morphine really rapidly and get a huge dose of morphine. I suspect I'm probably an ultra rapid metabolizer because I can take 1 cocodamol 8500, I'm asleep. But this is variable between humans. So this is the sort of thing where we can start looking at this to predict how people are going to respond to pain relief. So that one is not currently in practice in the UK, but this one is. This was the flagship test in pharmacogenomic test introduced in the UK. It's DPYD. It's a test that looks at an enzyme that breaks down fluoropyrimidine chemotherapy. So that's 5-FU, capecitabine, and Tegafur. Those are drugs that are used in cancers such as head and neck and bowel, used in about 38,000 patients per year in England. And the issue here is that people have variability in this DPYD gene. And if you have, because remember from earlier on, we were talking about you have 2 copies of each gene. If you have 2 low metabolizing copies, which is about 1% of the population do, you will not metabolize this enzyme very well at all, and you're at risk of death if you're given the drug. You can have 1 high and 1 low metabolizing copy, and then you might get given a dose reduction or a reduced dose of the drug to reduce your risk of side effects. Or if you have 2 regular metabolizing copies, then you'll have a normal dose. It's not to say you won't get side effects, but you're not at risk of this huge risk of death that you have if you're a very low metabolizer. So this is a medicine safety issue. And we have a new pathway in place that's gone in for a couple of years ago, where you have a test for DPYD function before you get prescribed the anti-cancer drug. And then patients at high risk of toxicity can be also an alternative or a lower dose. So we're looking at data at the moment, but the idea is this is going to reduce the severe toxicity and risk of death. So there are 4 commissioned pharmacogenetic tests at the moment, which doesn't sound like very many, bearing in mind there's good evidence for drug gene pairs, maybe over 100, probably up to 400 there's some evidence for. We only have 4 currently commissioned. So DPYD that I've just mentioned, also MTRNR1, which was mentioned earlier. Karen talked about this. Is the one, if you remember back to talking about risk of hearing loss with aminoglycosides. So 1 in 500 of us carry a variant in this mitochondrial DNA gene that confers increased risk of hearing loss when given aminoglycoside like gentamicin or amikacin. So it's commissioned for people who have long-term conditions that predispose them to getting aminoglycosides, such as CF. The downside to this is, because you think, oh, well, we give lots of gentamicin to people who are septic. The problem is this takes 4 to 6 weeks to turn around. So, of course, it's not actually any use in the acute infection setting, but hold that thought, we'll come back to that. So we're currently only looking for 1 variant in that gene, but there are 2 new variants being added. So that's the first 2 that commissioned. And then we've got TPMT and NUD-T15, which are tests looking at metabolism of 6-mercaptopurine and azathioprine. We can already do TPMT biochemically, but this is a genetic way of looking at it, and it's only used in ALL, so not for those drugs used in any other setting. So those are the 4. That's it. We also do have HLA testing. I mentioned this in the context of abacavir. So this is done through the tissue typing labs, and so is not in the National Genomic Test Directory. These 4 are in the Genomic Test Directory. You can go and have a look at those if you want to. But yes, so testing for abacavir, carbamazepine and allopurinol, particularly in the context of risk of Stevens-Johnson syndrome, you can do that. And it's particularly recommended in patients from certain ancestries who have a higher risk of Stevens-Johnson's and toxic epidermal necrolysis with those medicines. So here's a picture of the National Genomic Test Directory, which you've seen already, and there's the R65 aminoglycoside risk of hearing loss and the commissioning statement. So if you want to go and have a look at the commissioned populations, you can go and do that. So that's what we've got at the moment. But what's coming next? Well, I mentioned that the use of that aminoglycoside risk test is not helpful in an acute sepsis setting. But there is now a machine that will do this in a rapid point of care test manner, and it takes 26 to 30 minutes to do. It's only been trialled in neonates, and this is in Manchester. I think they had 1 or 2 patients in Liverpool, but the majority were in the Manchester neonatal unit. And this was appraised by NICE, not under their technology assessment programme, but under their diagnostics assessment programme. And this was published in the 30th of March 23, so earlier on this year. And the recommendation was that this could be used in the NHS, but further data needed to be collected because it was only done in a big tertiary neonatal unit. So they want to see if it works to the same efficiency in non-tertiary neonatal units. And also this doesn't come with any funding. It's not commissioned. So the funding needs to be found locally for this. But that is an interesting development, and you're likely to see more point of care tests coming through for genetics. So what else is happening? We have a national pharmacogenomic project called Progress, which is being headed by the Northwest GMSA. So we're in Central and South, our neighbours up to the Northwest, running this project, which is looking at a pharmacogenomic panel test. So multiple pharmacogenes on a panel being administered via a general practice setting, or being administered at a general practice setting. So they've done everything from looking at the best test to use in the lab through to the IT requirements and the flow of data through the system, which is a really important thing. And looking at patient acceptability and what happens with the data. So they're hoping to roll this out into the other GMSAs next year, so it might be coming to a GP practice near you. Also, this was a landmark study that was published earlier on this year. It was a big European study across 7 European countries, including the UK, 12 gene pharmacogenetic panel, 18 hospitals with community health centres and community pharmacies as well. So they were able to show that using a pharmacogenetic panel to guide therapy, so this increased dose, reduced dose, or avoid altogether, using that kind of pharmacogenetic intervention reduced adverse drug reactions by 30%. So this is sort of showing you that it is possible to do this and reduce adverse events. There are some caveats with that paper, which if you want to ask me questions about, well, we can talk about later, but it's really important to show that it can be implemented in lots of different settings and it can work. And also, it's also worth knowing, and we've had a mention of this earlier, that online providers are starting to offer direct-to-consumer pharmacogenetic testing. What I would say is it's a little bit buyer beware, because there is no regulation of this. So therefore, if you have a genetic test done for pharmacogenetic variant, are they looking at all the possible variants or just the 2 that they want to test? If there's 136 and they've only looked at 2 and they say, oh, you're negative for those 2, what does that mean? So there are lots of things to think about. We are working nationally, sort of pharmacists working with GPs, working with British Society of Genomic Medicine, to look at a statement around how healthcare professionals should and could respond to being presented with direct-to-consumer testing. There is an existing statement from the GPs and the genomic medicine people from 2019, which you can go and Google and have a look at, but we're updating it to include more pharmacogenomics. Also, one of the big community pharmacy chains, even Day Lewis, has launched a community pharmacy-based pharmacogenetic panel test, which they are working with their local GPs. If anybody's seen it, please let me know, because I haven't actually seen a practice yet, but that's on their website. You can go and have a read about that. Also, this one is likely to come soon. The expected publication date is to be confirmed, but this is another nice diagnostics assessment panel investigation into genotyping cytochrome 2C19 for patients who've just had a stroke and are about to be given clopidogrel. So why am I worried about 2C19? Because clopidogrel, again, is a prodrug and it doesn't work as an antiplatelet if it's not activated by cytochrome 2C19. So looking at the activity of your 2C19 from the genotype is going to help us decide whether this drug works for you, because 1/4 of the UK population is projected do not activate clopidogrel. So this is just looking at this in people who've just had a stroke. But of course, we use clopidogrel after heart attacks and in peripheral vascular disease and lots of other settings as well. So this could potentially affect a lot of people. So stroke, for instance, caused 116,000 hospital admissions last year. So that's the number of patients we're starting on clopidogrel pretty much universally for that. So this is going to have a massive impact on practice potentially. The draft recommendations came out in June 2023 and included a point of care test. There's another one available apart from the Genomics one now. GeneDrive have made one, but also potentially offering lab testing. So we're waiting to see what the recommendations are on that. Also, this is a drug that I wouldn't expect most people to have heard of, and I don't mean to be patronising by that, but it's for a very rare indication, symptomatic obstructive hypertrophic cardiomyopathy. It's about 6,000 cases per year in the UK, but this is a nice technology appraisal. So this had 90 days to be implemented from the 6th of September, and under the dosing requirements, you now need cytochrome 2C19 status to be determined from genetic testing. So this is going to start imminently and is likely to be done in association with the drug company, is my understanding, but ultimately may go into NHS testing. So if you want to find out some more, because we've had a bit of a whistle-stop tour through pharmacogenetics today, but there's a lot out there that you can look at. Here's a lovely QR code you can scan, which will take you to the Genomics education programme at what was called Health Education England, but is now NHS Workforce Training and Education. They have so many fabulous free courses, resources, everything from sort of 2-minute bite-sized things you can watch up to hours, and podcasts, and there's all sorts on there, including some of the teaching slides I've used today. They're on there. So do go and have a rummage. Incredibly high quality, can't recommend them highly enough. This team also sponsor in-person training, so modules on the MSc Genomic Medicine, 2 certificate, diploma and MSc in Genomic Medicine. They sponsored me when I did mine. Also, there are some massive open online courses on MOOCs on FutureLearn, which are typically free for a few weeks. And if you want to keep them for longer, you need to pay, but that's enough time to go through them. And also CPP is one of the pharmacy providers who have a nice introduction to genomics in pharmacy and pharmacogenomics. And the last resource I'd mention is Genotes, which has already had a plug today, but they did launch a pharmacogenomics module a couple of weeks ago. So please do have a look at that. So what do I want you to do? Well, I don't need everybody to have an MSc in genomic medicine and be able to do all of the pharmacogenomics and be incredibly whizzy about that. But I do think we all need to have a basic level of genomic literacy so we can start having these conversations with patients and signposting them to the resources that are available. But the good news is there's plenty of free training available. I think it's worth having a think about where pharmacogenomics could impact on your practice. I haven't come up with a specialty yet where it's not going to make an impact. Even if you work in IT, which you might think is far away from patients, we need the genomic data to move effectively through the system with the patient and to be surfaced in the right way. So this is really important. And the GMSA are here to support and connect you with the right people. So please do keep in touch with us. I think that's been a theme that's running through today. But there's my email address if anybody would like to ask me any questions. I'm going to stop now. So thanks very much for listening.
Wow, thank you so much. I always like listening to your pharmacogenomics talks. They are fantastic. To everybody that's still on the call, I really hope that you've really benefited from this webinar because, as Hayley said, it's about getting that awareness of genomics. That's the point of these webinars, and thinking about how genomics is now impacting us all in our practice. So I think because we are running over time, we're going to do some questions, and because what I'm going to do is go back to front basically and start from questions for yourself, Hayley. And sorry, my chat is not working very well. So I've got the first question is from Lucille, and she wanted to ask about the TPMT. She says, is that not for IBD patients yet? And if when will it eventually happen?
Yeah, it's a really good question, and it is specifically only commissioned in ALL at the moment. I think these things are constantly under review, and so, you know, it may well come eventually. And a bit about this is the capacity of the labs to test everybody, and so we need to make sure that we have enough capacity in the labs to do so. What I would say is anybody can apply to get a test put onto the National Genomic Test Directory. Anybody on this call could do that if you want to. So you can go to the National Genomic Test Directory. If there's a particular test you think should be put in for your patients, then you can make an application and put the evidence forward, and then it's assessed by pharmacogenomics, the National Test Directory Working Group, and that includes pharmacogenomic tests. So I'd encourage you to have a look at that if it's something you're interested in doing for your patients.
Thank you. I just have a question as well. Is that are genomic data going to be put on all pharmaceutical labels in the future as pharmacogenomics becomes more part of everything? And does that mean then who's doing the consent to do that genetic test for the patients?
When you say labels, do you mean the leaflets, the leaflets that go into the packaging? But yeah, those leaflets replicate the summary of product characteristics, which is effectively a license for the drug. We are at a national level working with the MHRA, who are the people that license medicines in the UK, to standardise the genomic requirements for information within those product information leaflets and labels. Also, because it needs built into drug trials as well. So currently there are probably between 70 and 100 drugs that have some form of genetic testing recommendation. I'm not going to say requirement because there aren't that many that are absolutely you have to do, but there are a lot where it is a possible thing that you could do. So it's really incumbent on us when we're helping our patients to understand what's happening with their medicines to make sure that they understand if they've had a test, what that result means for them. So that there are various examples, and I mentioned a couple of them, carbamazepine, for instance, in patients from particular ancestral backgrounds, they're at higher risk of Stevens-Johnson. So we should be doing a test on them to test for particular gene sequences. But yes, it's really incumbent on us all as healthcare professionals to support our patients to understand what they're doing with their medicines and their treatment generally, but particularly medicines. I think it's very easy for patients to think, oh no, it's far too complicated, I can't possibly understand that. But it's our role to help them understand that and simplify it for them if necessary, but also give them a true choice by giving them information that is pertaining to them.
Right, thank you. And I think that just goes to show how important it is for us to all increase our knowledge and awareness of genomics, but also it's also knowing who to signpost patients to them when they have these questions. So thank you so much. Has anybody else got any questions for Dr. Wickens? Okay, so I'm going to go up. Michael, are you still on the call?
Yeah, I'm still here, Phil.
Okay, so there is a question here for you. When you're doing your talk, it was, how do you deal with non-paternity results? Is this something you communicate with patients, the patient or parents of a patient?
So that is a very good question. And there is no sort of a single straightforward answer to that question either. I think the first thing to point out is that in genetics, we're not in the business of breaking up families, first and foremost. But I did have some discussions with a little while back with Annika Lucassen, who's a professor of genomic medicine at Oxford and who's been heavily involved in the genetics forum and things like that about this. And essentially, there's no clinical national guidance over how these should be dealt with. So it's very much deal with locally on a case-by-case basis. And also there may be other things that factor into it, like how important that say a mismatched paternal sample, how much a father sample would actually be useful in the analysis. That might affect how you play it. There were a number of mismatches that came back during the 100,000 Genomes project. I know certainly for a number of these, a quiet word might have been had with, say, the mum of a proband, and just is there a possibility that maybe this person is a dad? And actually wasn't uncommon for the people to turn around and say, oh, actually, yeah, I could have told you that. Which again, I think highlights the importance of stressing this at the start of the test, because actually in those situations, it was clearly, if the parent had actually understood why we used parents from both samples from both parents, and it needs to be their biological parents, then you wouldn't have gotten into that mess, and we would have gotten all scared about having some difficult conversations. So that really, I think, just highlights the importance of having those discussions early on. But like I say, there's no specified approach, so it's very much case by case. But I don't know if that's really answers the question or not, but
I agree with you. It is a case by case, but as you quite correctly said, it is thinking about those conversations you're having when you're doing a consent conversation and explaining about the test, and also explaining that they will be looking at paternity. So I think as long as you've given that clear explanation and people are making that informed decision to go ahead with the test, I think that's one of the ways. But you're right, it is a very tricky conversation. And I suppose one of the first things you also have to make sure that the samples haven't been mixed up.
Yeah, definitely.
Yeah.
The other thing I would say as well is was particularly important, I think, with the rapid whole genome sequencing pathway for acutely unwell children, which is if we don't know, it's a very bespoke pathway for children in NICU and PICU, where a diagnosis is going to give you an answer, change your treatment plan. So there's a sort of 2 to 4 weeks turnaround time for those. And actually, for those families, it's really important, because the last thing you want when you've got a child who's in NICU is to then find out you've got a paternal mismatch as well. That's not what that family needs at that moment in time. So again, all the more reason to have that conversation. Yeah.
Are carriers for CF to make them eligible for pre-test screening counselling?
No, anyone can, if there's a family history and someone wants to explore their risk, then they can be referred to genetic counselling, and part of that will be considering whether they want to be tested, if there's a test available in their particular situation. So it just emphasises really the importance of
good quality genetic counselling, and then having it at an early stage. I mean, many pregnancies aren't planned, but in an ideal world, you know, people would consider their family history before conception and have that referral before they conceive.
Great, thank you. There was a question right at the beginning about the record of discussion that needs to be signed when you consent in a patient for whole genome sequencing, and I think it's Jana had asked if it needed to be signed. Can it be scanned and sent electronically? And yes, it can, but also remember that that is the patient record, so it also needs to be uploaded to the patient's EPR. So I think those are all the questions. I do apologize if I've missed any questions, but I just wanted to say thank you so much to all our speakers. You've all been fantastic, really great information that you've given. And to everybody that's joined the webinar, I hope you have got benefit from joining the webinar and have got a greater insight and awareness into genomics. And as we said, this is a series, so we have had the first one, and that is on the YouTube channel. This one will be uploaded to YouTube as well, and we will be holding our third one on the 29th of February, which, whilst it is also leap year, it is also Rare Disease Day. We'll be sending out information about that closer to the time. But thank you to everybody. And with regards to the GMSA team, I don't know if anybody wanted to add anything. Charlotte, Donna, Karen?
I was just going to say what a great morning. Thank you for comparing, Phil. That was brilliant. All the speakers were great. Everybody was engaged. Thank you for all the questions, and we look forward to seeing you next time.
And
Yes,
I've just put something in the chat saying thank you to all presenters and to everybody who's attended. And just said any feedback, very much appreciated.
Yes. Thank you all. I think we can all go and have a late lunch, and apologies for running over time, but sometimes it's difficult to stop when a talk is so interesting. Thank you very much to everybody.
Thank you very much.
Thank you.